Guys, let's bring on the lovely Danny. There he is. Hi, Danny.
Hey there, how's it going, Balint? Let me make sure that, let's see, that I can hear you.
Test, test.
There we go, that should do it. As that loads, and unmute site. Okay, good. No, wait, no, I don't want that one. Hang on. That's better. I'm still hearing myself. How did I do this before? Shoot.
It feels like it's a mystery every time, right? Because the settings keep changing even though we set it.
Exactly, yeah. I don't know why that is. How do I mute this?
And Chill, welcome in. On the Diagonal, welcome in as well. Yes, it is time to cross over with Professor Anduza of Paleontologizing.
I'll be back.
Thank you, Tommy.
Oh, I know what's going on. There we go. That's a lot better.
Awesome. Danny,
Sorry,
thank you so much for coming on again and for our
of course.
the weekly crossover. This is very quickly becoming my favorite night. It is just so much fun to chat about these different fields that I'm not readily exposed to, and get your thoughts on these, and get you in a room and we get to talk science. It's a blast, man. Thank you so very much.
I completely agree. I don't know. Back when I worked at museums and back at university, I got to talk science pretty much every day with colleagues. I miss doing that, so this is really nice to have that. It's very healthy for me. So thank you for the opportunity.
No, same here. It's been missing, and it's cool to be outside of our field and chatting.
Yeah,
and,
for
like,
sure.
Right, I would never have read this paper otherwise, but I'm like, man, these are really cool studies, and it's, you wonder then like how these like approaches could inform.
This is true.
Something like also in our field and vice versa. So it's like, like with the genetics, like maybe we can't do the genetics experiments on the dinosaurs, but you start wondering like, from an ecological perspective, would they have had these behaviors and effects that we're studying in simpler organisms, and how that might all tie together. And it's a fun thought exercise, I think, too.
It is pretty cool, yeah. You're kind of getting at the fundamentals in paleontology, like where you work with what you've got and you just kind of go from there, So yeah. That seems to be a continuous theme with these so far, is like you're just going, how can you be content with so few data? But it forces us to get creative, and I really like showing that off, and that's one of the reasons why I picked this paper. This is such a creative way of looking at this problem, like how nutritious were these plants that these dinosaurs were eating, and I kind of wish that there were some figures included in this.
Yes,
I'll tell you about them, but there were some, like, I don't know how familiar you are with how they're able to extract these gastric juices from the cows in order to run these tests.
No, so I was reading the paper, and so Chad, they're trying to demonstrate how certain plants and foliage can be broken down by simulating, is how I took it, as like the gut of herbivorous animals, and then collecting the gas that comes off this digestion process as a metric. Detector, welcome in. We got a rating from Detector. Detector, how was your stream? I hope it was lovely. Folks, our friend Detector does leatherworking, also physics streams sometimes, and games. Welcome the heck in. We're near. Our raid is moderate to severe. Detector, welcome the heck in. Raiders are having a special crossover stream with Professor Anduza of Paleontologizing. His name is in the description of the episode today, so click the like and subscribe button on his name. Drive him on over, folks. He does this full time. Like, go sub to him. He's on that Partner Plus push. Go do all the things for Danny. Y'all, welcome the heck in. It's great to have you here.
I appreciate that, Balint.
Absolutely. So Raiders, we're talking about this really cool study where I'm having trouble like understanding even how to begin to visualize how this experiment was done, but they're trying to demonstrate how well plants are broken down by different animals. And if, trying to have, if I'm understanding correct, Danny, using modern day plants as a proxy for what existed in the past during the time of dinosaurs.
Yes.
And seeing how well these modern ones can break down in the guts of animals by simulating a gut environment. And that is collected by the gas that comes off of this breakdown, and like the amount of gas that comes off is a proxy for how nutritious it is. Is that an apt description of the experimental design?
It is, yeah. I think that's really good, first of all. So nicely done. I know these papers aren't always the easiest to understand, but yeah, you got that. One thing that I want to emphasize, I guess, is that we're looking at dinosaurs from the end of the Jurassic period. So like dinosaurs like Allosaurus, Stegosaurus, Camptosaurus, Apatosaurus, aka Brontosaurus, dinosaurs from this time, from the late Jurassic. Trying to figure out the big plant-eating dinosaurs, how could they possibly survive on what people used to consider to be like, very kind of poor plant material, so not very nutritious. The really important thing to understand is that a lot of our modern plant groups had not yet evolved. We didn't have things like fruit trees. We didn't have grasses. We didn't have flowers. The angiosperms, the flowering plants, did not exist yet. And so these are our floras. These are like plant communities that are dominated by conifers, like pines and redwoods and things like that. Ferns and horsetails, club mosses and spike mosses, stuff like that. The plant communities would have looked really different from today, but everybody always kind of assumed that like these are not plants that are very nutritious at all. How in the world did you have the biggest animals that ever walked the earth surviving on these? And this is a really cool approach to that problem.
And so, a naive question is, the reason why they're testing for those particular dinosaurs is because afterwards you had a population of different kinds of plants that are considered traditionally more nutritious? Okay.
So during the early Cretaceous period, so not too long after this time, the age of the dinosaurs goes Triassic, Jurassic, Cretaceous. So at the end of the Jurassic, a bunch of dinosaurs go extinct, a bunch of plant groups go extinct too, and then you get new plant groups evolving during the early Cretaceous, and new groups of dinosaurs evolving alongside them. And that's when you have your flowering plants first develop. So the first flowers show up in the early Cretaceous. And so prior to that, we just had things like gymnosperm plants. So like non-flowering plants. Ginkgos, cycads, horsetails. Horsetails might not actually be gymnosperms. I don't know my plants as well as I should, I feel, but
But it's definitely those that have even in modern times are considered as least traditionally nutrient poor. Like everything that you're listing off is like not, you wouldn't imagine a large animal heavily eating it, or almost like horses eating low quality hay. It's just like they're just eating it, and the amount that they need to eat for it to be nutritious is either like borderlines on the absurd, which is why you give them additional supplements to offset the deficiencies.
Sure. Yeah. Lita would probably know a lot about that, given that she's worked with horses and other animals.
So. That's the only reason why I know that as an example.
Yeah. But these were the only plants that were around for these dinosaurs to eat. So, like, how in the world are they surviving on that? On things like ferns.
And we actually, I think Hanny's kind of leaning on some of the, like, with the paper title is, do we know anything about the bacteria in the dinos that would have had in their stomachs, like the gut microbiome interaction?
We don't. Oh, and hopefully you can't hear that. Thank you for the follow, whoever that was. My audio isn't coming through there, is it?
No, but we can, guys. Please, we can't do a fancy shout out yet, but please, his name.
It belongs in a museum.
His name is down below. You can easily click it. Like when you click on, guys, I will put up the page and show you. Like you don't even have to click out of the stream. The heart is right there. Easy peasy lemon squeezy to click. like the reason we are here on Twitch is because of this man, this science communicating legend. Please go follow him.
Oh, please.
And also, Danny,
I
thank you
appreciate you
so
going.
much for Sunday, y'all. Danny, like last minute, stepped up and hosted a redux
no
of the
worries.
science panel, and he just like on the fly, like I don't know if you were at all stressed, but like no stress on your face, made a new scene, pulled everyone in, and it was so chill and so lovely. Just thank you.
I'm glad it came across as running smoothly because it was, yeah,
It was
didn't feel that way.
no,
Yeah.
Nerdowino and I too were like, I don't think he's phased by anything. It was just so chill, and just thank you very much for stepping up, and it's just again,
You bet, yeah.
really appreciate it.
Anything for promoting science here on Twitch. That's the whole point. So, yeah. Thank you, by the way, for sending me some text to remind me, or emails, whatever. However you reminded me about this thing. Thank you for doing that, because there was a chance I would have forgotten about it. Been really busy lately. I'm still kind of.
Well,
in the
you're
process of
moving, and you're still moving your old stuff, and it's
Yeah,
hard.
yeah.
And then you got the furry overlord of like collecting rent. There's a whole thing.
Yeah, we'll see if any of my landlords show up on stream tonight. Like last week.
Yeah,
We'll have to see.
I just enjoyed last week. You're like, there's the landlord. Yep.
Yep. Yeah. So yeah.
But yeah, so I guess about the use of bacteria, and if we knew anything about that gut microbiome and how that could have helped contribute to the breakdown.
That's a great question. The thing is, we don't know anything about that. Like, we don't know what particular kinds of bacteria these dinosaurs had in their guts. What was their gut flora like? We don't know. We do know that they had big guts, especially the big sauropods, the big plant-eating dinosaurs with the long necks and the 4 legs. We know they had huge rib cages. They would have had lots and lots of space inside for like fermenting these plants. And that's what we think they were doing. That is like, they're what we maybe call hindgut fermenters. So the plant material's staying in those guts for a long time, and just having a lot of nutrients extracted from it.
That's actually what was.
What did those bacteria look like or anything? We don't know.
That's what was asked in chat too. So Smikes was wondering before you even mentioned it. That's the idea of the hindgut fermenter, and that's one possibility.
That's basically the idea. Yeah.
So as one thing that I actually would have thought interesting to see in this study, and I wanted to know how you felt. Like, would you think this was bogus? Is if we took modern day bacteria that we found in herbivores and put them in with the experiment that's going on here, is that at all some kind of marginal proxy to ask if there had been bacteria? Like would you take that as some kind of evidence, or would you be like, no, like this is too distant and the populations probably have diverged too far?
It's the sort of thing where it's the best we've got, so we got to run with it.
Mm-hmm.
There's that, and also one of the interesting things that they did in this paper is that they took different bacteria, different gut floras from different herbivores today, and they basically got very similar results from all the plants, whether they used bacteria from a cow or from a pig. And so that kind of shows that, like, it doesn't really matter the particular cocktail of bacteria that you have in there. It's like the plants are going to exhibit the same kind of signatures in terms of how nutritious they are, I guess.
Gotcha. Okay.
Yeah. Maybe there were wildly different gut bacteria back during the Mesozoic, but we basically don't have a way of testing that idea at this point, and so we're doing the best we can with what we've got today.
I mean, like you've said, though, we've talked about ginkgos, right, are very similar. Like your modern ginkgo, like what's around outside, to what the ancestral state was. Yeah. I love your wall, by the way. Every day, it's like slightly different. I love the rotating exhibit. It feels like it's there.
I appreciate that. I wanted this to be totally modular and just be able to change it out every day. And so it takes a lot of work to do that, but I'm glad you appreciate it.
Every day I'm like, all right, come on,
I really like
it's the background. It's awesome.
Yeah, and then I got my Christmas tree over here too.
The little Charlie Brown Christmas tree.
Yeah. But yeah. Anyway, what were you talking about?
So I guess like if the trees that we're using are very similar to what they have.
Oh yeah. So ginkgo in particular.
Yeah.
That's actually a genus name. Ginkgo biloba is the genus and species.
Yeah.
And that's the last surviving member of that entire group. They used to be all over the place, really, really diverse. Now there's only one genus and species left. But that species or that genus has been around since the Cretaceous, I think. I want to say it goes back like almost 100 million years. So it would have been pretty similar to what we had back then during the Jurassic.
And my naive guess is maybe the bacteria that would help break it down are doing something similar, or at least if not the same, genome, the processes they're using to break down the plants. If the plants themselves haven't changed that much, then presumably the bacteria breaking them down haven't either.
It's like this idea in paleontology where like the players can change, but the game stays the same. Like you've got different organisms that they might look different from one another, they might come from different lineages, but they're doing the same kind of ecological job, and so they evolved to be pretty similar to one another, even though they might not be related.
Yeah.
And that's probably the case for the bacteria, I would imagine.
Yeah, which I think that's a pretty cool way of thinking about how this works, right? It also I think gives this really unique spin of what's going on with this particular paper. Like being able to, like, that's why I thought it was so cool, because I was like, I feel like you can use this and like it makes good sense to me, but I'm like, I need Danny's expertise on it because I don't I don't have the training for it, but it seemed like it was a really clean cut study on that front.
Yeah, and one of the reasons I picked this is that I actually saw the lead author of this give her talk at the conference that I was at back in June in Salt Lake City, the Mesozoic Terrestrial Ecosystems Conference. So there are all these paleontologists and geologists from around the world who'd come from as far as Australia, South Africa, South Korea, and China in order to present their research and just kind of commune with other geologists and paleontologists there. And it was a ton of fun. This is maybe my favorite talk from that. Mariah Howell, I met her at the conference, and she did this work at the University of Bonn, I think, in Germany. It's so cool to finally see it published because the talk was really, really neat. And it was super fun talking to her about that after the talk. And I'm just glad that it's out there now. It's been peer-reviewed, it's been published. This is cool stuff. This is really cool stuff.
Yeah, I was surprised that the method was published a while ago. It said back in 2008.
Yeah.
And it just seems like such a cool method. I'm like, I don't know, I guess we'd never maybe see it in our field, but it was just super unique. I feel like there's an opportunity, though, there, too, where I would have thought it would be nice to combine, because they were collecting the gas, right, as a means by which to measure how good the bacteria or the breakdown product is of the plant. There's that mass spectrometer machine that you also, works on this gaseous state, and it would actually be able to analyze the compounds. And so it would have been cool to see like what the metabolic byproducts are based on the breakdown via the bacteria. I know like the price would skyrocket, right? Because the samples are so pricey, but I thought that would be like a really cool like next step almost as I was reading this. Like, oh, that'd be a cool collab.
Yeah. I know during the talk, the author was showing just photographs under the microscope of this plant material after it had been digested. So like kind of lining up these different types of plants all in a row so we could see how much they'd been digested. And that was really surprising too, because some of the results were not what I would have expected. Some of these plants were actually far more nutritious than you would ever imagine. Things like horsetails, which we think of as very like abrasive, like really silica rich, horsetails have been called scouring rushes because native peoples would actually use them to like wash dishes, to scrub pots and stuff like that, because they are so abrasive. And but it turns out they're actually not that difficult to digest, and you can get a fair amount of energy out of them.
Wow.
And I was kind of blown away by that.
It's interesting, actually. Lita with the horses, she's trying to weed up all the horsetails because they're just like she's like, I don't want the horses like near them because they're not nutritious and they're abrasive and all. Exactly what you were saying. That's a really interesting take. I don't know if the horses can digest it given their how the digestive system works, but
and horses are finicky like that too. Like I've read that horses are like physically incapable of vomiting. So if they eat something that's not good for them, like it's not coming back up.
Yeah, it's surgery.
Yeah. Ugh. But also horses chew their food. So these dinosaurs were not doing that. They were just ingesting that whole. And so maybe all the silica in that would be really harmful to a horse's teeth. But horses also eat grass, which is also very abrasive and very hard on teeth. So they'd probably be fine.
And that's what Hogan was asking. That it's my basic understanding of digestion that chewing is the first part. Wasn't it important for some dinosaurs evolved teeth for chewing vegetation? So would that have been after the angiosperms?
There weren't a lot of dinosaurs that focused on chewing until the Cretaceous. You've got a few that pop up here and there in the Jurassic. Things like Camptosaurus and Dryosaurus, they chewed their food. But you don't really get the big-time dinosaur chewers until the Cretaceous. And it's around the same time that angiosperms evolved. And so those two things might be linked to one another. But if we're thinking of our big sauropods, the big plant-eating dinosaurs with the long necks and 4 legs, they did not chew their food at all. Here, I can show you, actually. Right here, we've got a juvenile Diplodocus skull, and you can see it's only got teeth at the very front of its jaw right there. There's no chewing teeth in the back. There's no teeth in the back at all. So these guys were just using these teeth to kind of nip off bits of plant material, and then they're just swallowing it whole.
Wow.
Yeah. These guys are probably feeding at ground level most of the time, if not all of the time. And they have those really long necks, not for reaching way up into the treetops, but for eating things like ferns, club mosses, horsetails, stuff like that, more or less at ground level.
Why do we think
those are some of the plants that were tested here.
Why do we think it's at ground level?
Well, for one thing, these guys have got really long necks, but the neck seems to go straight out in front. It doesn't seem like they're adapted for reaching up into the treetops.
Gotcha.
Other dinosaurs like Brachiosaurus or Camarasaurus, they've got necks that are built for reaching up high like that. That's kind of the natural posture of their necks. Diplodocids like Diplodocus, Apatosaurus, aka Brontosaurus, they're not built for that. They're feeding at ground level, we think. And they're just using that long neck to kind of feed very efficiently. They can sweep their neck in a big wide arc without having to walk around very much. So it's like a very energy efficient way of feeding too.
Okay. That's cool.
Yeah.
I also love the form as function, that you can look at the teeth and you can look at the skull, and that, like Smikes was saying, it's a little bit similar to like modern day cows with the positioning of their teeth, and actually gain it like insight as to how their feeding behavior was. Like that's really cool.
Exactly. That's the kind of paleontology that I do, Balint. Yeah, that's like my particular research is a lot of form as function stuff. Functional morphology is what we call it. But yeah, like my work on Spinosaurs is in that same vein.
I'm waiting. I'm so excited for that paper.
Yeah. Because I'm hoping I'll have time to work on it after, sometime early next year. Oh boy. The move is taking longer than I thought it would, and I've got some other stuff going on too, but
trust me, publications take forever. I just remember the very first time I saw your channel trailer. You remember there's that line in there about you're working on Spinosaurids. Can't tell you about that yet. I was like,
I still can't.
Professor!
years later, yeah.
And that's just, that's just how it goes, right? Like we're always...
That's science for you.
Yeah, like the review process, like all of it just takes forever. And so, y'all, that's not just me being like giving Danny a hard time. That's just us being like this. It really does take a very long time to have these papers published. And that's not a fault of the researcher. That's just like finding it a home and formatting. And it's a
lot of work. And revising the manuscript because it's taken so long to write that new papers are coming out. Now you have to cite those, and you have to change what you're saying, and you have to incorporate this new work. And oh man, it just kind of snowballs like that.
The Tradoon,
especially on the Spinosaurs, because there's always new things about them and new publications.
Hopefully, when it comes out and you come on, I'd love to just chat with you about that paper and just get like
That'd be great.
some of the secret insights. You know, like things that were. I think it's when like we interview each other about a particular paper, and like you're the one who wrote it and you did the research, and it like you don't think about a particular detail like as a detail of it. It's just kind of like, you know, floated by, and then it was like, oh yeah, I got this idea because of this like weird one-off thing, and it's just like the story behind the paper is half the time as fun as the paper itself.
I totally agree. I'm looking forward to that now, Balint. That's gonna be really neat talking about that.
We have a couple of questions about this. So Golganek asks, is there any thought of using the digestive juices and microbiomes for modern birds for similar research as well? So I guess, are there birds that eat ferns and horsetails, or is it just like our cows and other?
That was like the first question that I had for Mariah, for the lead author of the paper, when I was talking to her about this. And she said, she's like, that's a great question, but it's outside of our capabilities right now. There's very few birds that actually eat, like, leaves. Folivores is what we call them. Folivory is leaf eating. And there's one bird that I can think of that does that as its primary diet, and that's the Hoatzin. You know about the Hoatzin, Balint?
No. How does one spell that?
It's Hoatzin. They are super, super cool.
This bird is so cool
right?
looking of a bird.
Beautiful, beautiful bird. And one of the coolest things about them is that the juveniles have claws on their wings that they use for climbing.
Wow.
And so kind of like their theropod ancestors. The young do a lot of climbing before they can fly, and like to avoid predators, they'll actually drop out of trees down into, because they live in like really swampy areas around the Amazon, and so they'll like kind of swim around in the water, and then they'll climb back up the tree using those claws. And then they later lose the claws as they mature. So it's a good example of like changes in ontogeny as well.
They lose the claws. That's interesting.
Yeah,
I wonder if
the claws just get resorbed. They don't fall off, but they just like slowly shrink and get resorbed into the wing.
So probably some age-related gene expression that has that happen. That's really cool, that transition timing between the two.
And I love that that's what you jump to as well, because I'm not thinking on that level. So that's really cool that
That's why I like this too. Like our thoughts jump to do very different things, and it's always fun to be like where our brains end up, and then chat. Because that's similar to there's transition states between other animals as well, like not necessarily physical changes but behavioral changes, and that's all like changes in gene expression. So, I'm assuming there's not a lot of genetic work being done on these because of money reasons, I would assume.
Yeah.
But it would be interesting
they're not
to
as
know.
well studied as they should be for how interesting they are and how unique they are among birds. Because they're also the only bird that I know of that's folivorous, so they eat leaves. Most birds cannot digest cellulose.
Yeah.
But Hoatzin can, and that's the primary part of their diet. They mostly eat leaves, which is.
Do we know?
Strange.
We might, or I don't know. But if there's like a bacteria that helps, or do they have a natural?
There's got to be, yeah. There's got to be like a particular gut flora that they have. I'm guessing. Like it would stand to reason that would be the case, right?
Yeah.
Yeah. So I asked Mariah about this. It's like, have you looked at Hoatzin gut flora, and like, would it be possible to look at that? And she's like, we don't have any Hoatzin in Germany. So, but what they did have was cows and pigs, and in her talk there was a really interesting image that she had that kind of gave me the heebie-jeebies a little bit, as a bleeding heart vegetarian, to see an image of a cow with like a big hole taken out of its side, and there's basically like a gas cap on it, and these are cows that they have at the agricultural school there, and or like for agricultural studies or something. But basically there's like a cow that just has like a bionic, it's got like an insert in its flank, and you just unscrew the cap and then you can reach your hand in while the cow is like looking around and eating and doing whatever,
and it's totally fine.
That's how they actually extract the gastric juices. That's how they do that. And I presume they have pigs that work the same way too.
And so it's just they just take the juice out and then put it onto the foliage that they're testing. In I was did you know what the machine looked like? Like I have these just naive questions of even like the experimental setup in terms of an image. You know.
I think there were different beakers that had plant material in them, and then the gastric juices were put onto that, and then that's hooked up to a machine that's measuring gas output or something like that. It was like Hohenheim gas experimental interface or something like that. It says it in the paper. I forget.
Yeah, it's the Hohenheim is actually a molecule as well in genetics, which is Mark's DNA, which is why I was just like, wait a minute.
Interesting.
Wait a minute, I don't think that's what this is. And
do you think it's named after the same guy, maybe?
No, it's named the original is also a German fella. But it's originally the Host, Hoerc
gotcha.
stain, and then it's been devolved into this similar.
It's a homonym, I guess.
Yeah. And I was just like, wait a minute, who is this?
Hmm.
But yeah, we had another question earlier in the chat if we know, I guess, do these birds that you were just telling us about, do they also have, I don't know, it's like stones in their stomach to help the breakdown of these plants, or do we know if dinosaurs did that?
Yeah, so I think it's no on both counts.
Huh.
Yeah, so I don't think Hoatzin have what we call gastroliths, stomach stones, for digestion. I don't think they do. They're birds, so they're not really going to be in the business of like swallowing big heavy rocks because they still have to fly. It would be disadvantageous to them, I would imagine. Birds will swallow grit and stuff, but anyway. But that is an idea that's also fallen out of favor for dinosaurs too. For sauropods, there used to be this idea that they were swallowing these big stones that would roll around in their gizzard and would, mush up the plants to help with mechanical digestion. It's like, well, since they're not chewing with their teeth, they don't have chewing teeth, they're chewing with their gut instead. Like physical breaking down of this plant material using stones that are rolling around in there. That idea is really, within the past 10 or 20 years, has really fallen out of favor with paleontologists. Where like we'd always kind of taken it for granted before, and like if you find stones near a sauropod skeleton, it's like, oh well, those must be gastroliths. It's probably not the case, because for one of the reasons why it's no longer the case, why we no longer think that that's true, is that the stones don't look like gastroliths in modern creatures, where they're really abraded and everything. And they've got like characteristic wear marks on them and stuff. They're all scuffed up in a really particular way. The gastroliths, supposed gastroliths from sauropod skeletons, are just really smooth. They look like river stones. So it's completely the wrong kind of wear signature. In fact, they probably were just river stones.
That's what I was going to ask. Like what do you think the stones actually were? They were just happenstance there, and it was like jumping to a conclusion.
Exactly. Yeah. Oh, and I'm getting a pizza delivery right now. Holy cow. Look at this.
I'm on the stream.
You are on the stream? Yeah.
Hey, it's a Lord delivery.
A Lord delivery.
Yeah.
I appreciate that. Thank you, Lordy. Holy cow. I was getting really hungry, so I appreciate that, Lordy. Excellent.
It's my fault. I'm keeping you far too late. And
no, you're great.
You should be eating dinner and things.
I got pizza now, Balint. I can't complain. This is excellent. I'm happy.
Guys, please follow Lordy. There's shouted out in the chat. She's feeding Professor Anduza and making sure he's surviving. Please drop her a follow.
Yeah. Cheers. Yeah. So, if you're wondering, like, how do you get stones like that, maybe in the ribcage of a sauropod dinosaur, if it wasn't swallowing them, how did they get there? Some of them may have just been ingested accidentally, maybe, since these are dinosaurs that are feeding at ground level. Maybe they'd eat a rock every once in a while. But what I think might be more likely is that, especially if it's in like a fluvial environment, if you find the skeleton in sediments that were laid down by flowing water, like a river or a stream or something, you're going to get river rocks in a stream, and they're going to move along until they can't anymore. And if you've got a big dead sauropod that's basically blocking the stream, you're gonna have a bunch of rocks and crud and just roll up against it and get stuck. You're also gonna have a bunch of sticks and branches and vegetative matter, but that stuff rots away. The stones are what's left. And so, yeah, that might be why you get stones in the rib cage of these dinosaurs, or near the rib cage, because like that's the biggest part of the animal, and it's just there in the middle of the river, basically making like a temporary dam.
So is this similar to the concept we talked about in our very first stream about wanting to name extra species? Right, that there's a school of thought of like everything that you find that is different, you want to jump the gun on a species, and so you see some rocks and you want to jump to a phenomena versus being a little bit more conservative about it. It almost feels like those school of thoughts.
It's, I don't know. I've never made that connection before, but you know, it would be a little bit of, I guess, like we would say in science, arm waving. You know. But yeah, nobody's really like stopped and tested that idea thoroughly until recently.
And this paper here kind of further goes what you were talking about, that it really isn't, it like you don't need the rocks to break down, right? Because my understanding is like they're just putting the gut juices on some plants. There's no like breaking the material. It's just like, here's the plant, put it on, as you're simulating how the animal would have eaten it, right? Because you're saying front teeth maybe to grab the plant, and that's about the only damage you're going to cause to it. No hind teeth to chew it. So you're just swallowing them whole, and then just put on the gastric juices and see what happens.
Yeah, and then these animals had enormous guts too. I mean, shoot, like the size of a large car maybe. That's a lot of space for fermentation in there. And it could have been that like when these animals were younger, they didn't have as much fermentation space, so they needed to eat, more nutritious plant material or something. But then the bigger you get, it's almost like a feedback loop. It's like the bigger you get, the better you are at eating, and the more space you have to ferment this stuff in your guts, and it kind of becomes more and more efficient that way.
Do we have any soft tissue evidence of gut structure and number, almost like, you know how a cow has those different compartments? I'd imagine those soft tissues are going to be difficult to fossilize, but is there anything to tell us it's one giant stomach versus subcompartments? And
we don't have that for sauropods, for the long-necked plant-eating dinosaurs. We do have some fossil guts from theropod dinosaurs, from some of the meat-eating dinosaurs. There's a little theropod named Scipionix from Italy, and it's like this long. It's like the front half of the animal missing the tail. It was a baby. It was a juvenile, not full grown, but it actually has preserved guts in there. So there's like this, almost like a carbon film, where you can see the viscera inside of the body cavity, which is really interesting. But I don't know if that's. I think the idea is that it's kind of bird-like, but it's also kind of crocodile-like, which is what you would imagine for an animal like that.
Okay.
Yeah, but for plant-eating dinosaurs, I don't think we have anything similar. I don't think we have any soft tissue stuff like that for plant-eating dinosaurs. It's been speculated that they would have had these big, what are they called? CISA or CISUM or CCA or something, but they're like big chambers to hold bacteria, basically to like cultivate a gut flora in there. And they, every time I see them illustrated, it's almost like they're like balloons that a balloon artist would use, where they're like kind of long and thin. They're like sausage shaped like that, I guess. And they're kind of like arranged around the sides of the stomach and near the intestines and stuff. But cool. I don't know if we actually have any hard evidence of that. I kind of doubt that we do.
So that ties into a little, we had a question from Smikes, which I think we've kind of been touching back and forth on this. So, I mean, as you were saying, like how your work is, you know, you're taking all these bits of evidence from modern day animals, the dinosaurs, and like piecing it all together like a puzzle. And there's not necessarily one picture of the puzzle like taken from the like zoom out view where you get all the evidence at once, but it's like this modern day, this ancestral state to kind of build the picture. how does peer review work on something like even this paper here? Like, I think the evidence is very straightforward on how it works on your modern samples, but when you're speculating as to how it might work on the dinosaurs, how do you have to write your paper to be like for us in genetics, like you always get slammed for overreaching too, right? And that's like, so how does one write and expect a review for something like this? And what is your boundary for when can you make your conclusion, if that makes sense, when you're trying to piece together so much?
It's funny that you say you get dinged for overreaching, because that's kind of what paleontology is. I mean, we're trying to reach beyond what we have and grasp at any bit of evidence in order to try and piece together what these ancient ecosystems were like. So I guess for a paper like this, I guess we could talk to the authors and see what that was like. But I guess it would get sent out to reviewers maybe who, I guess we could probably look at the review history. But there's probably some reviewers who are in like the agricultural side of things, like actually looking at digestion in modern animals. We've probably got some paleontologists as reviewers, I would imagine, but I'm not entirely sure. Yeah, it's tricky. This is kind of an off-the-wall paper. Like, this is not the sort of thing that I would typically read. This is not just a description of a fossil. This is not like a really phylogenetics heavy paper or anything like that. And I think that's one of the reasons why I found it so interesting.
It's very unique for the papers that we've looked at or that you've talked on yours. Like, it's a very much a blend of modern techniques to inform the past.
Yeah.
And not like the phylogenetic side of thing. That's why I was like, that's why I want to start with this too, because I'm like, this is a really cool paper. But yeah, it's interesting. I didn't realize that PLOS One had the review history and comments uploaded nowadays. But I didn't realize they started this in August of last year. 2022 was the first submission.
Huh.
And so I'm surprised that's the very first time that it's actually that's how long it took to get it published, because it seems really sound, like, I guess at this stage, and with 6 reviewers. Like, it had a lot
6
of
reviewers. Holy cow.
Yeah.
Yeah.
And so it was just it seems like a lot, and it's very just interesting to have this. I guess Julian Andres Castigo Vargas was one of the reviewers.
Vargas sounds familiar. Yeah.
Yeah, the other ones wanted to stay secret.
Gotcha. Okay. Interesting. Yeah.
It's kind of cool. I'll put it in chat, y'all, if you want to also read through it. It's also in the Discord, the paper, but it's a neat feature. Like, I know eLife was one of the first ones that started publishing the review and responses along with
text of the reviews.
Yeah. And it also gives you some accountability. I guess, I mean, here you're still shrouded with anonymity. Like, you can still say, no, I don't want to say who I am, but at the same time, I don't know, maybe knowing that it'll be public, you don't want to be as much of a jerk.
That's generally the everybody I've heard that advocates for the idea of open review like that, that's usually their primary reason, is that it guards against people just acting out of hand, being a jerk in the reviews.
But I guess it's interesting about how the review process is so different between our fields too, and like you said, it's just the nature of the field, right? It has to be different. It's just like, when you explain things, there's such a nice logical step-by-step piece to it, and I'm just like, not all the papers that we've read together have that kind of built in. And maybe part of it is just that there's assumptions built in the paper, and that you don't need to spell it out for a non-paleontologist. But it's just some, like, this one to me didn't seem jumpy. It seemed to me like this is something that is, you're looking at ancient plants, that we know
Yeah.
don't evolve fast, that we know are not very different from when they were 100 million years ago.
At least phenotypically, they're not. They're very different. Yeah.
Yeah. So and then I feel like the assumption of the the microflora hasn't evolved. Like maybe they on the whole genetic level they look a little different, but the process that breaks down something that's also that slow evolving, presumably it's not an arms race, right? They're not building up against one another and trying to like fight off the other and back and forth. It's just they're just there.
For most intents and purposes, the plants we have today, like ginkgos, cycads, horsetails, they're going to be pretty similar to the ones that existed during the late Jurassic, and the gut flora, for most intents and purposes, are going to be the same thing too. So, yeah. Those are the kind of baseline assumptions that we have to accept before we can do science like this. And, I think as paleontologists, we have to, we got to be cool with that.
I mean, but it doesn't seem like a stretch to me. Like this one totally like I'm like
I'm glad.
I'm like I'm fully on board with this. This one is and like Smike said too, the title of the papers too is great. I can understand what they're trying to say. That's awesome.
Not too much jargon in there.
Yeah, it's because there's a lot, right? There's
paper,
a lot of
really
papers in both of our fields that are just like if I haven't been working here a decade, I'm not going to know what you're trying to say, and I feel like it's accessible, it makes sense, and it's an exciting, straightforward title and study.
Right.
Is there let's do a couple of other questions here in the chat. How would you say, from Hogan, does the evolution of teeth relate to the digestive process?
Oh, so dinosaurs that could chew their food, there wouldn't be as much incentive for them to get super gigantic, because it's not like you have to have this huge fermentation chamber in your gut that like gets more efficient the bigger it gets. So that's probably why ornithischian dinosaurs, the bird-hipped dinosaurs, never got nearly as big as the sauropods, just because they didn't need to. There's not that feedback loop that's like pressuring them in the direction of getting bigger and bigger and bigger. But also the evolution of teeth would allow dinosaurs to feed like not the evolution of teeth. Dinosaurs start off with teeth, but the evolution of chewing teeth like that would probably allow them to maybe not feed for as much during a given day. But that might be completely wrong, actually. If you have to sit there and chew your food, maybe it does take longer. I don't know. Just kind of grasping at straws here, I think it probably would influence the kinds of plants that you can eat, and maybe allow you to eat things like tree bark and stuff like that too, which we have some evidence that that dinosaurs like hadrosaurs were eating stuff like tree bark. The duck-billed dinosaurs were doing that. Were you present for that, Balint, when we were talking about that?
So,
like, why in the world are these dinosaurs eating rotting logs?
It was a few weeks ago, right?
I think so, yeah.
Yeah. It was, you're like you're saying, it was the duck-billed platypi.
Or the duck-billed dinosaurs.
Dinosaur, not platypus, jeez.
Hadrosaurs is what we call them, yeah. Anyway, but we think that they were doing that to get to the invertebrates inside.
That's right. It wasn't that they were digesting the bark matter. It was just chewing through and a byproduct of being there. Because that's something that's a mystery for researchers today, is how on earth termites break down bark. And that's like the only it hasn't been discovered. Like, we don't know yet what the mechanism of it is. There's two reasons to study termites. One is to break down the bark, and two is their epigenetics. Because when they hatch, they're this nymph stage that can become a soldier, a worker, or something else in between. Crux Gaming! Welcome in, Crux. Welcome in, Raiders. Crux, how was your stream? Tell us all the things. So the Raiders coming in. My name is Balint. That's Professor Danny Anduza. And we're having a crossover stream chatting about paleontology, genetics, and more. Crux, welcome the heck. And I hope you had a great stream. If you were crafting something cool, feel free to share it in the Discord. We'd love to see what you were doing. Chatting right now about this really cool paper about the breaking down of plant matter in the guts of the animals, and we did a quick rabbit hole into breaking down bark and tree matter, and talking about how the only two things that you study termites for right now is how you break down wood, like what is it in their gut, and then that epigenetic information of them becoming, like, from the nymph stage, a nurse, a soldier, and then a potential reproductive as well, which is unknown how that pathway marks. And it seems to be different from ants because they evolved sociality independent of bees and ants. So it's only the second time that you had eusociality in insects. And so it's this very unique piece. Did a keychain and bracelet for Peace Initiative. Very nice, Crux. Very, very cool. Thank you very much for the raid, guys. Go check out our friend Crux Gaming TV. So Danny, is there anything else that you would want to highlight in this study that you think is, like, super, super cool?
I just think it kind of challenges some preconceived notions about plants during the age of dinosaurs being really crummy. Like this idea that like, oh well, you know, they must have had to eat 24/7 in order to be able to survive on such poor forage material. That doesn't seem to be the case. So it kind of makes things make a little bit more sense that these dinosaurs are able to grow so very large. And I also think it's really interesting too which plants turned out to be more nutritious, like horsetails. I never would have guessed you could make a living eating horsetails, and yet this demonstrates, like, got a fair amount of nutrients in them. Cycads were not a real surprise. There's not a lot of not a lot of nutritious stuff to be had in cycads unless you're eating their fruits. But where's my cycad? Here, I've got one over here. A cycad plant, not the fruits. The fruits are super toxic, and they will hurt you or kill you if you eat them. But this is some people at home might be familiar with this kind of plant. This is Cycas revoluta, which is a plant that's really common in cultivation. Cycads used to be super, super common in a bunch of terrestrial ecosystems. Now there's only a few species left, but this is like a classic plant from the age of dinosaurs. People say it looks prehistoric. And yeah, look at all those gifts. Holy cow.
Unarmored, thank you for the 7 tier 1 gift subs. Armored to HLT3199, mid-tier gaming, Rogan 27, the Brian Boru, Bioerotica, Engineer Ober, and Scotch Down Under. Unarmored, thank you for those 7
nice.
tier 1 gift subs. I appreciate the heck out of you. So I tell you, some of the names throw me for a loop. I've had ones where they come in and it's clearly a troll name, and it doesn't register until after I've said it out loud, and there's been 6 clips, and I'm like, oh, they got me on that one. But Unarmored, seriously, thank you for those 7 gift subs. I appreciate you. And Grimly, welcome the heck in. I hope you're doing well. Hanny says, I heard that there was a time when tree bark could not be digested by anything. Maybe the bacteria break it down is just in the environment.
Yeah, there is an old idea that, like, the reason we have so much coal from, say, the Carboniferous period is because the fungi that break down dead plants had not evolved yet, but I don't think that's actually true. I think that's kind of an antiquated idea that's fallen out of favor recently.
That was just actually put in the chat, Danny, as a potential hypothesis. So it's interesting.
Oh, really?
That's no longer the in vogue idea.
Yeah. Yeah.
Nice.
Anyway, cool paper. Thanks for talking about it.
No, this is I was reading this. I was like, man, this is really cool and very, very different. I was very excited to read this, and yeah, thank you for that recommendation. Shall we move on to the next topic?
Let's do it. Yeah, let's do one of your papers.
Do you want the aging or the magnets?
Let's do the magnets one first.
Okay.
Yeah.
Go ahead and grab it. All right, so this paper, y'all, there's a lot of cool things happening here. First is that there's evidence for this transgenerational inheritance of a behavioral preference. And the reason I thought this was cool, and I assume that there's no way to test this in a dinosaur, but I wanted
Hello
your
there.
idea
Hello there.
on like if you thought this
Hello
is
there.
a possibility on something.
In dreams, one
Whiz
is
Kid,
not
hello,
tethered by
good night.
earthly limitations.
What does that mean?
Come.
So this term is one of those jargony terms that I think makes sense after you talk about it, and it's not like this transgenerational term, because apparently there's a lot of deep-rooted definitions that are not apparent on this word. So what it means is that it's an inheritance across multiple generations, from parent to offspring to that offspring's offspring and so on, without there being a genetic mutation, but there just being some kind of change in gene expression.
Okay. So
And
that'd be genetic in that sense.
Yeah, but what's weird about it, and the reason I always point this out to folks is because we got scolded by a reviewer for this, is it's transgenerational if it goes for multiple generations.
Okay.
And it's intergenerational if it only goes one.
That makes sense. Okay.
So
Okay.
So what's already cool in the paper is that they were talking about that it's you're spanning multiple generations of a behavioral preference. And why this is so cool is because fruit flies, if you could, there's an easy trick you can do to tell if you have a brand new fruit fly, and you take a vial and you flip it upside, you put the flies in this vial or container and you flip it upside down. If they go from the bottom to the top, they're young.
Is it like senility that prevents the older ones from doing that?
It's they seem to have a lack of preference for going against gravity as they age. It's not just it doesn't seem to be just linked to muscle degradation. So like just it's not that they can't move as well anymore, which is the easy thing to say, but rather there is an actual young fly preference to move up against gravity.
Interesting.
And so whenever you have like a container, right? Like if this is the top, if you tap the flies down, like you hit it on a desk, they come right back up. You know, they'll be up here. You turn it upside down, and then they go right back up. And you can just keep doing this. If you wanted to, like the first time I saw this, I did this for like 20 minutes straight. They just kept going over and over. And so it's this very reproducible, natural behavior. It's not just in lab populations. It's wild caught. All the insects do that, right? Like with respect to fruit flies. Yeah, and it's
my mind immediately jumps to the question: how can you tell that's a preference that they have and not just an ability or lack thereof?
So you mean it's like an ability for detection?
So how do you know that the old ones just are not incapable of doing that? It's just that they don't have that preference anymore.
I see. So there are tests you can do for like locomotor activity, and that's the big catch in doing with the older flies, right? So you want to ensure that they can still move, and it's not they just like grow, like you're saying, like they go really tired or their muscles have broken down, and it's not that like you said they don't have the preference, but they just don't want to go against it. So what you can do is actually you can slow down or speed up aging in particular tissues of interest in flies. So if you have, let's say in the leg muscles, you want to age those, you can turn on genes that produce free radicals and age that particular tissue, but leaving the brain in that young state, right?
Interesting.
They actually do that to ovaries in the fly to look at chromosomal segregation. So like how there's an increase in trisomies in humans. If you age a fruit fly ovary, you start getting reproductive errors as well. South Rebel, thank you for the one I'm not here biddy. Thank you, South Rebel. So that's one way to do it. And when you do that, you accelerate the aging in these muscles, but leave the brain. You find that they still will go, they'll negative geotax, they'll go up against gravity. It's just slower. So the young-brained animal still has this built-in preference that's different from the old animal.
And you can tell that the old animals, it's not like their ability to sense what's up and down has diminished. Instead, it's definitely just a preference that they just prefer to be up more when they're young.
Yeah, what's cool about this study is that I think they very nicely asked that question, right? Because the whole basis of this study is they starved flies, the moms, and they put a magnet near the food. And so what they're actually going is they're going up against the opposite direction of where the flies would normally go. So instead of negative geotaxing away from the magnetic source, they're going towards it now because of the food. And so what they find is the offspring go against gravity, or go towards it, I guess, go downward, in spite of you giving them the stimulus that's where they should go.
Gotcha. Okay.
Yeah, so it's passed on, this preference, to the next generation, even though that generation, those F1 flies are never undergoing that starvation stress. They're never taught that you should have this preference, but it stays. And what's weird is it stays 5 generations. And the reason that's weird, Danny, is because there have been other organisms in very different transgenerational effects that have been studied, and the number 5 keeps repeating.
Interesting.
Like nematodes, and they pass on an immune up regulation after a stimulus that decays with time, but it's 5 generations. Lita's PhD work was addiction behavior in fruit flies that was passed on, lasting 5 generations. Here again, it's this number 5, and that there's enough of evidence.
Yeah, that's interesting.
Yeah, it's like there's enough evidence to suggest that it's real, and there's some pattern to it. But what it is and why it is, no one knows.
That's really cool. That's really cool.
Yeah. And so I was kind of surprised by the study in that they looked at male. They had a nice figure of how they did the experiment, right? They had the exposure with the starvations, and looked at like the larval adult state when they did the collections, when the preference showed, and this is the big one in panel F. Right, they had a couple of non-significant ones, but here, this blue one, right, that's the mock exposed, and here's the one with the magnetism. You can see that geometric score of going, not negative geotaxing, but going towards the gravitational source is huge up in blue versus the sham. It's, it's by chance, it's nothing really. So it's...
Sure, yeah.
Yeah. And yeah, Jim, how molecularly it counts generations... With Lita's study, we found that there was a particular neuropeptide that was turned off in the next generation, and then with each subsequent generation, it was still off, but a little less off. Right, so at least on one molecule what's working, but like what's turning it a little back on, and how that dial works. That's the big like unknown part of it.
Interesting.
And Laura Brandett, is it possible to tag fruit flies? Yes. So you can, it's not as beautiful as you might think. You can put a drop of paint on them, and that can mark them. You can also glue a QR code on the back of them. So you can have cameras that can track the QR code. There's also...
It's gonna be a tiny QR code. Holy
cow. Oh, yeah. But there is this really amazing software called Deep Lab Cut. And so here's an example of a figure. Hold on, let's open the... So this is a software that can track the animals, and it can actually tell the difference between different organisms. So it's a machine learning technique. It's images that learn, like the machine learns like this image series is from this animal, this is from another. So if you have a video camera over these flies or mice or honestly anything, you can track its movement. And what's cool about this software, it's free, it's open source, and you can train it on anything in the world. If you can see a difference on it, you can use this software to get a numerical amount of difference.
Holy cow.
Yeah.
What I wonder is, Balint, could you use that software for that thing at baseball games on the big Jumbotron where they've got like the 3 hats, but you do the shell game like that?
So
Would it work for that or no?
I think it would. So there's actually a lot of really cool examples here. I think this is the page for it. Maybe. So locomotive activity. So you can see how it could be very quickly
cool.
applied to being used on like the movement of the bins. Horse
That's
racing
awesome.
and horse gait behavior. Danny, here you go. You're gambling.
Wow.
Whisker movement. Pupil
Very cool.
pupil dilation, swimming behavior, and it's computationally intensive. You need a very powerful graphics card because you're analyzing
Sure.
video. But the idea is that you teach this algorithm on a few videos, like you label the dots and you tell it, okay, this is this thing, this is that thing, this is this X, Y, and Z, and then you give it brand new videos afterwards, and it's able to impute all those dots.
That's very cool.
Yeah.
Holy moly.
So Lord Brandad, they didn't do that in this particular paper. If you wanted to do individual tracking, they even had. Oh, I think this was on the previous page. Here we go. Thank you, WizKid, and thank you for the follow, Jaimin Way. Here is a fruit fly that's being tracked, and it's actually, you can track the egg. I don't know if you can see. There's a little dot on its ovipositor when the egg gets laid, and even the egg has a dot on it, so you can actually track egg laying events.
Very cool.
Yeah, and it's you're really limited by your imagination using this software. I was using it for ant behavior and trying to categorize what is a particular type of ant behavior and getting all the numbers out for it. So like, here is a leafcutter ant soldier. It normally does these behaviors, and when we change the genes expressed in its brain, now it does these behaviors, and now you have quantifiable numbers, and you can actually do the compare and contrast, and it's not just a by eye, well, it did a little more here versus here. You can assign that
That's really
number to it.
I wonder what you would have to do in order to basically like break this program. Like, at what point would it admit that it's made a mistake? Like, if you were to create a video, a fake one, with things that are actually identical to one another, like, how identical would you have to make things before it'll admit, like, oh, it's broken, it's not working. I can't track these things because they're too similar.
Like when it's multiple animals?
Yeah.
It breaks if you have the animals constantly leaving and coming back into screen. So if, for example...
So it's just tracking them, it needs to be able to track each of them.
So like in this example here, you see how at least 2 of the 3 are always on screen. And one might leave screen, but when it comes back, it still has 2 of them on. So you have built in as one of the parameters is 3 total. But if you were unbiased, like just wanting a total, like here's 100 plus or minus 30 that are constantly coming on and off, then it would break.
Interesting. Okay, that makes a lot of sense now.
Yeah, there's definitely those limitations of it, but it's super powerful, and it's free. I'm like, wow, that's
really neat.
Yeah,
that's really neat.
It runs in Python for those interested.
Was this created by somebody in a laboratory, or is this just like a programmer did this? This is presumably through public funding. This
Germany.
It's really cool that it's free.
Yeah, a German lab.
Germany.
Yeah, actually, a husband and wife team of scientists made
That's
it.
Super cool.
Yeah, and so
there's going to be statues erected of them, I'm sure.
They are, and they're really nice people. They are really nice, like, which is like a nice, it's like, no, they're not like, I did this thing. No, they're just like, they messaged me and was like, oh, how can we, like, because they knew I was using it because I tweeted at them like, look at cool thing I did. Then they messaged me like, how can we help optimize it? And I was like, oh, okay,
awesome.
sure.
Wow.
And so I set the parameters, and they're like, here's what we would tweak, and here's how the numbers we would tweak for it, and they even have this. I think you'd like this, Danny. It's called the Deep Lab Cut Zoo. And so they're having citizen scientists do labeling on videos of different animals, and then uploading it to a database, so that let's say you had a kangaroo and you didn't have necessarily the computing power to make a new model. You can download the modeling that it's already been curated and run it against your video on like a regular laptop CPU and track the behavior.
That's very cool. That's
Yeah.
very, very cool. Holy moly.
And yeah, I thought that was I'm like, that's really cool. And
Awesome.
there's so much going into the future of it. And yeah, so Nye, it works in 3 dimensions as well. So there's I don't know if they have the video, like the little preview up on here, but there are ones where it's like you're moving in X and Y and then Z with multiple video angles, and the software can impute all of those angles together to give you a dot moving in 3D space, which I think is really, really, that's like the next iterations that have been coming out, and it's a really powerful tool.
That's really neat. Oh, one thing I'm also curious about is, you said it needs a really powerful graphics card. How powerful are we talking here? Are we talking about like a gaming computer's graphics card, or are we talking about like something that's far and away more powerful than that?
So it works on an NVIDIA graphics card. So it for some reason, yeah. So apparently, like I knew this because Apple had NVIDIA for a long time, and we wanted to run it on a Mac, and we called them and we're like, hey, we just checking if you're using NVIDIA. And they're like, oh, everything we use works with NVIDIA. And they like promised us, and we got the computer delivered, and like 2 seconds in, the software was like, we don't work with this, we need NVIDIA. So it's like a gaming laptop, and we ordered like for the lab this custom computer that had 2 graphics cards talking to one another and just sped it up.
Nice.
But it wasn't like, you could use it for like regular gaming. It wasn't like this like insanely wild developed one. Yeah. And timers, you can use it on plants as well. I've seen it used on plants because like the leaves go up and down with circadian rhythm, and it's a way to do like screens as well. So if you have video cameras on 10 different mutant plants and you just want to see how the circadian rhythm changes, leave the camera on, measure the dot movement, and you get the output. It even gives you a default graph that you can look at, and you're kind of all set. Which I think is really neat. They didn't do it in this paper that we're reading today. The one with the gravitational pull. It's an older paper. They didn't have the tech out, but I always like the idea of like for future experiments, it would have been neat to utilize.
Right.
Yeah. So good rabbit hole
Neat
question. They didn't. So this was like the neat figure too, is that they sorted by sex, and they found that both males and females have this preference for the magnetism that's inherited, which is neat because there are plenty of other studies that demonstrate that it's sex specific. Like there's always will be one event, like with Lita's study, it was females, and it was passed through the female lineage. So exposure of the father did nothing to the female lineage. It was the maternal exposure that was critical. And here it looks like, maternal or paternal, it doesn't matter, both sperm and egg get affected, and that information's passed on to that next generation, affecting the sexes equally, which I thought was really
That's
cool.
really neat.
Yeah.
Yeah.
They didn't get to any genetics in this. So like what genes were modified and how they were modified. I imagine that'll be the next step, but I thought overall it was a neat study. And opportunity for us to talk about transgenerational inheritance, and I wanted to put you on the spot, Danny, and ask if you thought this was a phenomena that may have happened in dinosaurs, or is there any reason to think that this kind of thing wouldn't happen? Not necessarily with magnets, but just like that adaptation.
Well, with magnets, I think I've got more to say about that. But
okay, no,
as for
tell us.
Transgenerational inheritance, I don't really know enough about this to have an opinion in either direction. This is like so far removed from my area of research and everything that I've it's still sort of a new idea to me that you can have that kind of inheritance that's not coded for directly in a gene itself. But in terms of the magnetic thing, there is this idea, I don't think it's been thoroughly tested, that the reason that birds can navigate so well is because they have a way to actually sense the Earth's magnetic field, and that's thought to be true for birds, but also critters like testudines, like turtles, and that may be one of the ways that sea turtles are able to go back to the same beach where they were first hatched in order to lay their eggs again. Both turtles and birds kind of bracket the dinosaur family tree.
Okay.
So turtles aren't as close to dinosaurs as crocodiles are, but they're a lot closer than things like lizards. And then birds are living dinosaurs. So did dinosaurs, non-avian dinosaurs during the Mesozoic, did they have, a geomagnetic sense like that? There's no reason to say definitely no. And there's some reason to say, yeah, probably. So
is there evidence for like herd-like movement from one side to another in dinosaurs, kind of like migration patterns in birds?
There is, yeah. So from a number of different things, we've got trackways of dinosaurs that seem to show them, just kind of beelining all together in the same direction at the same time. So
what is a
moving in
trackway? Like what is?
Oh,
what
trackway
is
is just a bunch of footprints together.
Okay.
So yeah. They're making tracks along a way, I guess.
So like their trail is like stomped down almost. Like this is the route that we take a lot, and so there's a lot of.
Oh no, a trackway is just footprints moving in a direction. That's
Oh,
all it
okay,
is.
gotcha.
Yeah, it's just a generic term for there's a bunch of footprints that actually show an animal moving.
Okay.
That's a trackway. It doesn't have to be like a game trail or anything like that. It's not like that. And then also we know that a lot of dinosaurs exhibited what we call nest site fidelity. So they return to the same place year after year to lay their eggs. A lot like sea turtles and like a lot of birds do too, where they've got these massive nesting colonies, and they go back to the same place once a year, often traveling really vast distances to get there. So we've got good evidence that dinosaurs were doing that, because sometimes we just have like pavements of dinosaur eggshell where it's accumulating for a long, long, long time. And sometimes they're actually reusing the same nest structures year after year. They would dig out these earthen nests, kind of like I don't know, maybe it would look like a big salad bowl, but made out of dirt. And just year after year, they'd go back to the same ones. Maybe they'd try to get there first so they can get the best ones, and I'm sure they're improving them year after year and stuff too, but they seem to have used the same sites, even the same nest structures, year after year.
Do you find less skeletons at that site of the dead dinosaurs? Like, is that just a site for reproduction, then we leave, or is there no correlation?
Well, usually, if those sites are being discovered in the first place, it's because bones have been found there.
Okay.
And eggshell too. But sometimes it's mostly just eggshell, and in some nests, like there's maybe one skeleton of a baby that died at the site, and sometimes it's just all of them and they're all in there.
Mm-hmm.
Sometimes also all the eggs are in there not hatched. So like something happened, there was a volcanic eruption, or a river overflowed its banks or something like that, and all the stuff got buried. Yeah. So it really depends. Yeah.
So they could have used magnetic fields to also direct that movement.
If they're going to be navigating like that and going back to the same place year after year, then it would make sense that they had the same kind of internal, stuff going on in their brains, like homing pigeons or like sea turtles. Since dinosaurs are pretty close to them, especially birds.
Yeah.
It would make a lot of sense. Yeah.
That would be cool. Yeah, I mean, with insects, we know there are ones, for example, that like monarch butterflies that supposedly use the Earth's magnetic pole, and there's also evidence that bees as well. And it's the mechanisms aren't especially like satisfactory worked out between insects and birds, and like what the differences are even between there, like how much conserved, like if it evolved once and it worked, and maybe it evolved the same way a second time.
Does this go back to the base of Deuterostoma or something, like the common ancestor of vertebrate animals and insects, or is it?
Yeah.
Actually, no. Insects aren't even deuterostomes. They're arthropods, which is you'd have to go back even further. But
yeah. So like, did it evolve independently, or did the common ancestor have it? And so it's
Yeah.
just, or did we lose it in some sense? Like
Right.
there hasn't been a lot tested, like in as far as I'm aware, in like the bird literature of like anything as obvious as like this, the negative geotax and young individuals like immediately detecting just the pull of Earth's gravity and going against it. But so yeah, I just thought there was a lot of neat basic biology in this of the animals, but also like this transgenerational inheritance, which, as you say too, I think I still grapple with the idea that this is a real thing where it's like a non-mutational change, and that leads to inherited changes across
That's
generations.
reassuring to hear you say that, because you literally work on epigenetics. You publish on this, and you're still like, this is weird. How does it work?
Yeah, well, I mean, I think it's such a new because it's. I'm sure when you were in school, right? They said junk DNA.
Oh yeah,
Yeah.
of course. It's like, oh yeah, well, yeah, or like there's got to be some sort of gene that's coding for this, and we just haven't found it yet.
Yeah.
It's like there's just this faith in this idea that like everything has to be genotypic, everything has to be coded for by some gene, and yeah.
And it's like the
It's
fact that the structure of the junk DNA and how the DNA wraps itself around something is what's contributing to how it's transcribed. So if you change that wrapping, that's what's that state is what's inherited, and that's how you have this like being able to continue. I actually have a figure that I thought might be kind of cool to take a quick look at, if you don't mind.
No, go for it.
Let me see. It's okay. It's the packaging, and so this is kind of speaking to how this might be inherited. So you can have a state, you can imagine, so the DNA is wrapped around these balls called nucleosomes, and that's what allowed the DNA to be packaged. And actually when we were doing our cancer charity stream a few weeks ago, one of our speakers showed us this figure. So that is The Rock and Jason Momoa. And they, next to them is a strand of
baby.
But next to them is
Combine their genes.
Well, if no, then you'll create the ultimate human, right? But so it's in one cell. The DNA, if you stretched it out, it's taller than they are.
Combined or side by side?
Side by side. So it's like, Jason Momoa, The Rock, and a DNA strand. The DNA strand's the tallest.
Oh, okay. But what if The Rock were standing on Jason Momoa's shoulders?
I think that's about the height. That's about the height that they would be. The DNA strand.
Oh, okay. Wow. That's more impressive than I thought. That's really cool.
Yeah, so it's
Is
like,
The Rock
how does
very tall? I actually don't know a lot about him.
about
is
which
he
one?
considered a tall person?
About these two?
Yeah, are either of them considered very tall?
I think they're
Not
both
like
over
that,
6
just making
feet.
conversation.
I think they're both
Yeah.
over 6 feet. I mean, The Rock
They
is
look
right.
twice
Like
as tall as I am. Holy cow!
he cooks a lot of stuff, right? Because it's always you smell what The Rock is cooking, and
Oh.
every movie he raises his eyebrow, and you're like, I love this man because he raised his eyebrow. And
So
Momoa
he's
is
famous
just just goofy funny.
Okay.
Oh, Rock is
So
6'5".
comedian and a celebrity chef.
Yeah.
Good to know. Okay. Sometimes there's background information that I need to.
Well, yeah. I mean, if we're talking about environmental effects on genetics, you have to know the background. Like.
There you go. Yeah. Thank you for filling me in on that. Yeah.
I've totally butchered it. I apologize, chat. The Rock is great. He cooks a lot and he wrestles and movie star. Yeah.
Nice.
But so, yeah, it's all based on this packaging idea. And so
the point is that the DNA is all compacted like that. It's
to fit in a single cell because there's so much of it. And so
how thin is that strand then? If you could stretch it out that far.
So it's
got to be extraordinarily thin, like less than a micron thin.
Yeah.
Or something, right?
Yeah.
Yeah.
So it's I guess to give China, it's like thinner than your like an eyelash, like even thinner than that, right?
That makes sense. Yeah.
Yeah.
Well,
And
it
that's
has to be because your eyelashes are made up of cells, and your cells have nuclei, and
yeah, which you have that word, but it's just like a good image, right? I think is that thinness, And yes, Cricket, you are 6'5". You're amazing, Cricket. It's okay. And so that's how it fits right into your cells, is that there's these balls that are wrapped around. And so these purple dots here are the equivalent of these 8 balls that things are wrapped around. And so what you can have is a state like this, where in white, these are clustered together, these histones.
Okay.
And because they're so tightly clustered, you can't turn on those genes. Like, they're inaccessible for the transcription factors. But if you loosen them up a little bit, like with the purple site right next to them, you can get transcription factors to come in and turn on a gene of interest and make the RNA and make the protein. And then what happens is, with the epigenetic changes, different regions close and open as a result of these environmental cues. And that will then turn on and off particular genes. And what's weird is, normally when you have an offspring, the epigenetic marks are erased. And so you tend not to inherit most of them. But when the DNA is rebuilt, some of the marks are put back. And so that's the how these are inherited. Is you're not inheriting a mutation that permanently turns off the gene. It's the state of open or closed, which I think is kind of like, what's the mechanism of it? And it's not clear. It seems to be different based on the phenomena. So,
and this is the same phenomenon that's going on here, where that heritability goes for 5 generations. Something about this works until the 5th generation, and then it switches off or ceases to work or something like that.
Yeah, and it's you know, and this isn't like normally without an epigenetic effect, right? It might be that these area in white, it might be they open up when you need it, right? Like, there's an environmental stimulus, it opens up, you turn on the gene, and then it closes back. But it seems like certain stressors cause this conformational state to be inherited across generations. And what that 5 is magic number of, we don't know. Like, why is it this 5? And again, if it's just, you see it in one organism, you're like, all right, that's just a weird thing. But if it's in multiple organisms with multiple phenomena,
right,
there's something important to it, right? But not every phenomena is like that. Some is intergenerational. Like, we talked about the Dutch hunger winter, when humans, right, were starving, and their kids had diabetes. But when those kids grew up and they weren't starving, when they had kids, the next generation didn't have diabetes. So it's not they just had a mutation, it's just the epigenome changed. And so it's plausible all the way to humans. But that's that intergenerational effect that's only lasted for that one.
Interesting.
Yeah. So it's, I don't know, I thought it was a cool paper. It was, magnets, behavior, and epigenetics.
It is really cool stuff. Yeah. I guess the thing that I'm most curious about is just, like, what is the mechanism for being able to sense magnetic fields in the first place? I remember when I was a kid, there was some sort of animal documentary that showed, like, geese flying south for the winter, and it's like, oh, well, they must have some sort of magnetic material in their brain that orients them. It's like, that seems like a gross oversimplification of this kind of thing. But it's really cool that, well, that's still a mystery. That's kind of exciting, and that we still have yet to figure that out. It's like a really interesting phenomenon that a bunch of different animals exhibit, and we don't know how it works yet. Like, that's exciting in and of itself. But I think it's also exciting that there's the potential to study this using a model organism now, using fruit flies, and maybe that'll be how we finally figure this out.
Yeah, because I think, a difficulty would be to study the birds flying, right, and migrating, or even a butterfly doing a migratory behavior, because like, you have to track them. But if you can, this is a little bit more simple. It's just turning a vial upside down. I can see that as being a potentially easier, famous last words, at least thing to initially test, and then you can see, right, if the mechanism's conserved, you can move to the migratory animals and figure it out if it's that same nervous system. But Because otherwise, like, I've known a butterfly lab, and they did something similar to this, where they were trying to knock out certain neurons and genes that they suspected were, detecting the magnetic fields.
Sure.
But they had to tag this group of butterflies and then go down to Mexico and see if they could catch them during the migration.
Catch the same individuals?
Yeah.
Wow.
Or at least have some of them show up, right? Or none of them, but then it's, are they not showing up because they got eaten, because they just died? There's a bevy of reasons why they might have been thrown, not worked.
Exactly, yeah.
So there's also ones that do like RFID chips and tags and like more of like a GPS signaling so you can track, but those are, I think I've only heard of one lab that does it on that particular front. Yeah. And yeah, the climate change effect, Timbers, is huge on migratory behavior. That's another reason why monarchs are on the endangered list. And Hillbilly says that they found homing pigeons have magnetic material similar to rock in their beaks. They believe it's helped with sensing the magnetic poles. That's interesting, Hillbilly. I haven't heard that. Thought it was all like calcified, like bone structure. I haven't heard that it was something like rock in there.
Yeah, I didn't hear it was in the beak either. That would be new information for me. I always heard it was thought to be in their brain itself.
Yeah. At least in insects, it's like they have depending on which insects you look at, there's either this third eye called the ocelli or the fake eye that detects UV, and it apparently is linked somehow to magnetism, or like dragonflies that have huge eyes.
Wraparound eyes, yeah.
Yeah, like the upper part of their eyes detects UV and potentially these fields, and the lower part of their eyes detects like actual imaging. But more what the mechanism is, it's still at least in the insect world, and I imagine then for birds and others, it's still up in the air.
What did you call the third eye thing in insects?
So it's the ocelli. So it's
ocelli. Interesting.
Let me spell it. It's one of those words that I can type.
Sure.
Yeah, 2 eyes. That's what it is. And yeah, it's this cluster of these. Like for example, this particular one has
they're visible
3
on the surface.
Yeah, you should be able to see it. You can actually tell if the animal does a lot of flight. If it has 1, it's probably a land terrestrial insect. If it's got 3, it's probably going to be flying a lot.
Because it's operating in 3 dimensions
and just like they can do, like, if they're walking around and foraging, that could be something they can detect sunlight and be able to get back. But yeah, with flight, you need a bit more information because you can get lost easier. You don't have the pheromone trail to take you back.
There's vertebrate animals that also have got like a third eye, what we call a pineal eye. It's like tuatara are a good example of this. They're these really cool kind of lizard-like reptiles, and they've got a pretty well-developed pineal eye, but it's covered by skin. And we think it's for photoreception, for something to do with like telling what time of the year it is, like day length and stuff like that, so they know when to go into their burrows and hibernate. Or yeah. So it's interesting that I get it's totally different function, I guess, in insects. Probably not a homologous structure, I would imagine. Probably something that evolved independently. But
I would think so. Yeah.
Mm-hmm.
And Hogan's asking, is that eye like what iguanas also have?
It is, yeah. Parietal eye? Is it the parietal? I think in Sphenodon and Tuatara, it might be kind of near like the frontals. I don't know if it's near the parietals, but it might be. We usually call it the pineal eye in Tuatara.
Okay.
But yeah.
Cool. Yeah, the eye systems of these animals I always find fascinating because they're detecting different things, and even here on this one, you can tell the structure of the upper eye looks very different than the lower eye structure, and it's because they're detecting different things.
What kind of is this a wasp or something?
Yeah, this looks like some flavor of wasp. The most obvious one are like the dragonfly eyes. Oh yeah, here we go. These are like see, you can even tell different color.
Yeah.
Like those upper cells are detecting something very different than the lower cells, and it's just
interesting.
Naked eye you can tell. And then their ocelli are hidden away. They are right there, next to the antenna.
Very cool.
But yeah, they have it too.
Huh.
So yeah, that's paper 2. Should we jump over to the next study, the sad extinction?
Yeah, let's do that. I didn't have a chance to read through the whole paper yet or anything. Again, been really busy, but I figured this would be interesting to look at.
Let me open it up. Hi, JD. I blame it's clearly the guest star. It's clearly the guest star feature. It's clearly not my files are too large and they freeze everything.
Definitely not.
No. Are the eyes defined by being directly attached to the brain or just light sensing? That's a good question, Hanny. Like, what makes an eye an eye? Ooh. As in at least in insects, both the optic lobe. Hold on, we got 3D prints, right? I could be cool like the professor here. So these little rounded ridges are the optic lobes. The retinas will be on the outside here, and so you have cells going from the retina connecting to the optic lobe, which then connects deeper into the center of the brain. For the ocelli, those are connecting deep into the fan-shaped body of the brain, which is, again, like a visual learning and memory center. And so they all end up connecting, Hanny, to the brain, but that doesn't mean that if they're light sensing versus image processing, like both can end up connecting. It's like a planarian, our flatworms that we look at, those have light sensing organs. Those are its quote-unquote eyes, but they still connect to the brain. So I guess it depends on what you define as an eye as well in this particular context. What do you think, Danny?
I mean, my first answer as like a paleontologist would be, if we're going to talk about eyes and for it to make sense, they should be homologous structures. So like an eye in an insect should be like coded for genetically the same way as it is in a vertebrate. And I don't know if that's the case. They might be independently evolved, or I'm not sure. But I'm used to thinking of things phylogenetically like that, in terms of their evolutionary relationships. So like, did it evolve in a common ancestor and then was inherited by all of its descendants? Then I would say that's the same structure.
Mm-hmm.
But
that's a good point. It's just like, what is it? Colloquial science versus getting like more of these very particular definitions and terminologies. It's almost like our species argument, right? Of what exactly is a species?
Yeah.
And it almost depends on the population that you're talking to, like who the audience is, and when it comes to defining these.
Right. Yeah.
Nice. Okay, so Danny, take it away. What this sad paper?
Yeah, shoot. Let me pull this up real quick. Actually, having just finished my pizza.
I'll drop it in the chat as well for those. It is open access. They paid $10,000 for that.
Holy cow! Yeah. Let me find this in here real quick. But I thought this was really interesting because when we're talking about extinction rates, this is something that we as paleontologists talk about all the time. Like the rates at which different creatures are going extinct. What is the background rate that we would expect? How can we tell when there's like a biotic crisis going on? That kind of thing. And so in order to do that, you kind of gotta define your terms. So yeah, and I thought this was really interesting too because, Balint, you've heard of the idea of the sixth extinction, right?
Is that the current extinction that we're going through, or is that independent?
Yeah, like when all of these different species are going extinct due to human influence.
I was shocked about the number of bird species that is tied to humans in just the start of this paper.
Yeah.
Like,
it's like 1 in 9, 1 in 10, something like that. Because there's something like 9 or 10,000 species of birds alive today.
Yeah, well, it said
as many as 1,400 species being wiped out.
Yeah.
Mostly on Pacific Islands, due to humans spreading out across the globe. Like, that's wild. Like, to have like 15% of your species diversity just disappear in the geological blink of an eye. That's wild.
What wasn't clear to me, Danny, in this study is, are they including, and Murph actually grabbed where my brain was, are they including the release of domesticated cats in this metric, or is this something where we as humans have hunted over 10% of the bird
This would
population?
be, you wouldn't be able to tell exactly how these species went extinct. It's just like people show up and then they're gone.
Okay.
So it might be, a lot of it is probably going to be the other animals that we bring with us, and it could be diseases too. It could be rats are a huge thing, because a lot of these islands, you know, you think about places like New Zealand today, where there were no native rats, and then rats get introduced, and suddenly, bam, you've got all of these species of bird that go extinct because the rats are eating their eggs. They didn't evolve in an environment with rats, so they've got no defense against that. And yeah, same thing happened in Hawaii as well, where there was like a ton of extinction events, like a ton of instances of bird species going extinct when people first showed up, because they brought stuff like rats with them. Yeah. Even today in Hawaii, there are species of bird that are on the verge of extinction because of mosquitoes that people brought with them from elsewhere. And as the climate continues to warm, due to, you know, human release of greenhouse gases, temperatures are getting higher and higher on average year after year. And the birds that are existing up in the mountains where it's too cold for mosquitoes, those birds are being squeezed further and further, where like the mosquitoes can make it higher and higher up the mountain every year as the temperatures rise. So the habitat for these birds is decreasing, and the birds have no, you know, native immunity to like avian malaria or anything like that, and that's why the mosquitoes are such a problem for them.
Because they didn't need it, because there were no mosquitoes.
Because they evolved in an environment where that didn't exist. So there's a bunch of different ways that the arrival of humans can spell trouble for native birds. I guess that's the point there.
Yeah, and I guess Hanny's asking, how long would it take for evolution to help?
I mean, it depends. Longer than it's taking these bird species to disappear. Like that's the whole thing, is that like extinction and evolution are basically two sides of the same coin. So like if you're a population that's alive, that means that generation by generation you're going to change a little bit. That's evolution, you know? It's just, you know, offspring are a little bit different from their parents, and their parents are a little bit different from their parents. That's all evolution is. Just change in gene frequency over generations. And when you've got a bunch of individuals dying or no longer reproducing, so you don't have that change through generations, that's when evolution stops, and that's extinction. Is that when they can no longer have offspring. So yeah, basically you're either extinct or you're evolving. And if you can't evolve quickly enough so that too many individuals are dying and they can no longer replace, you know, they're not reaching replacement rate anymore, you have declining populations, then you go extinct. So I hope that answers the question. Shoot.
I feel like if their reproduction time was faster, of like turnover, that they could outcompete maybe.
Yeah, so creatures that do have smaller generational times, like for instance fruit flies or bacteria, where like, you know, it'll be a matter of days or hours, it's like generation times. They've got a certain degree of resilience, I guess, especially if they have a big population, because they can evolve pretty quickly. Whereas, you know, other species like elephants, it's what, 18 months for an elephant pregnancy? That's long generational times, and it might be years and years and years until they're able to reproduce. Maybe they're like 15 or something before they can start having babies. But the thing is, there's sort of a trade-off there that we see across species, is that like if you've got really short generational times, that usually means that you're really small. It usually means that you don't have a very big habitat, and maybe your whole species could get wiped out by something like, I don't know, a landslide or a volcanic eruption. Like your entire species could die out. Whereas for elephants, if there's a landslide, you know, they'll just walk over to the next valley.
Yep.
So like, there's a trade-off there. There's always a trade-off.
It's
And so yeah, with these birds, like if they've got very slow reproduction, that probably means that they're pretty large and they can move around a bit more easily, or they're like restricted to an island, like in the case of critters like the kiwi bird of New Zealand. They also have very long generational times, and they only lay one egg at once, and it's huge. And that's one strategy. But they've been able to do that and be successful in doing that just because they've been isolated for such a long time. I think that's true for probably most of these birds in the Pacific, and to a certain extent in the Atlantic and Indian Oceans as well. On these islands, like this is where most of those extinctions are happening, or on these islands where these birds have, they've been specifically adapted to a very specific environment, and it makes them more vulnerable to intruders, I guess, if that makes sense.
Yeah, I mean, the way I took it is there's the red queen hypothesis, right? Where if you have a predator or a disease or something that targets you, you're always trying to evolve a mechanism to outcompete it. Whether it be if it's like your receptors that the virus detects, right? You're trying to get your receptors to be really good to detect the virus to stop it before you get sick, and then the virus is trying to evolve a means by which to circumvent your immune system, and at the end of the day, neither are moving forward. You're just staying steady.
They're
but
running
here,
and running and running just to stay in place, basically.
Yeah. So it's like that Alice in Wonderland reference, right, to the Red Queen.
Mm-hmm.
But here, you're introducing like a new parasitic threat or disease state or something without there ever having been any evolutionary selection pressure on the population. And so I guess, there might be one individual on the island, right, that is a little bit more resistant than the rest, but the time at which that embryo is laid and it passes on to its next generation, like, it's just not fast enough. It's just they're not designed to counteract that. If they evolved with it, then presumably there would have been the ability, there would be that genetic diversity in the population to fight it off, but if there's no selection pressure for it, and speaking of trade-offs, right, that's something it's not an issue, it's a raid. Dr. NPHD, welcome the heck in, Raiders, folks. Dr. NPHD is a fruit fly geneticist and does microscopy and science here on Twitch. Go check out our friend Dr. NPHD, y'all. Raiders, welcome the heck in. How was your stream? Did y'all find any tardigrades today? Also, Dr. N uses Minecraft to build protein structures and visualize protein structures being built, which is really cool. Icefire, hi. Misadventures in Astro, welcome the heck in. Raiders, my name is Balint. I'm 1/3 of SciAnts streams. This is Professor Danny Anduza of Paleontologizing. We're having our weekly crossover stream chatting fossil science, ecology, evolution, genetics, neuroscience, epigenetics, behavior, pretty much what we separately specialize in, and crossing over and having really unique opinions about each other's fields and like asking questions and really pushing that dialogue or science as a dialogue. So welcome the heck in, y'all. Dr. N says, I always get jump scared by your raid alert. I expect it at this point, but it gets me every time. Today we're actually looking at a paper on how one frame shift in the COVID vaccine and how it's not a big deal and it's just cool biology. Nice. That does sound like really cool bio. Yeah. Well, Dr. N, welcome. And we're talking today about, we've done 2 papers so far. It's our paper 3. First paper was on the identification of how plant matter might be broken down, in particular during the time of the dinosaurs, like ferns. There's actually bacteria that can break them down, and they are actually much more nutritious than we originally thought. Danny, please correct me if I butchered the finding.
Yes, it works, yeah.
We also looked at transgenerational inheritance of behavior in flies via magnetic field. So here is that study, possibly. There we go. Published in, I know Danny's favorite journal, PNAS. And now we're looking at...
I always pronounce it phonetically, but yeah.
Yeah, I know. That's what makes me chuckle. And here is the current one we're chatting about human-driven extinction of birds. And it's it's quite shocking how much, like over 10%
Really
of
is.
all known birds have gone extinct as a function of human, I guess, colonization of islands that didn't have humans before, or the diseases and animals that we brought with them. As a microbiologist, trees confuse me. James R. Gurney. Me too. As an insect geneticist, I think trees are amazing, but also how they communicate is something also kind of over the top wild to me. You can have trees talk to one another about predator threats and it cause gene changes in genetic regulation. And it's, I always thought, is a tree actually alive? It doesn't move. Is it really sentient? And then it does all these things that are otherwise.
See, when you said trees, I immediately thought phylogenetic trees, and I'm like, well, yeah, of course they're confusing.
Oh, I don't know, James. Are you talking about like phylogenetic trees or like trees? That's what that's where my head went.
Yeah.
So Dr. N, that is a great question. We were actually just talking about that, of like what percentage of human extincted birds are due to us lugging cats around. So this data, as Danny was telling us, isn't broken down on, if it be cats or disease or rats. It's just us influencing, right, Danny, the living.
Yeah, I would imagine that rats would be a bigger influence, but also just like basic habitat destruction as well, especially like using fire to kind of change a landscape, which is something that a lot of early humans did. Like when you look at this diagram, people coming out of Africa and then into Asia and then down into Southeast Asia and Oceania, down into Australia, and then across the Bering Land Straits into the Bering Land Bridge into North America and South America. A lot of that really early on stuff is going to be just changing the environment with fire, and also like maybe driving mammal species into extinction, which also puts stress on the birds because those mammals might be like keystone species in those environments. Stuff like that. There's so many different ways that you can make things bad for birds, unfortunately, and so many different things that people have done over the millennia.
Which is an interesting term too, Danny. How do you describe a keystone species? Because I've struggled with this because it seems like if ever you take out an animal, it's gonna cause an effect in the web. And so, like, where do you draw the line of what a keystone is and what isn't?
That's an excellent question. Shoot, the way that I always learned it was like, they cause an outsized influence on like the structure of the ecosystem. So it would be like sea otters. They keep the urchin populations down. Otherwise, the urchin will overgraze the kelp, and the kelp provide the habitat for all these other animals. So it's like you pluck that out, and then the whole thing collapses. But I'm not. I'm describing the concept of a keystone species, and you already know that, Balint. But like, at what point do we draw the line between, like, would the urchin not be a keystone species? Would the kelp?
Right? Yeah, exactly. Like, do you call the kelp also, but then the urchin that like keeps the kelp in a certain amount of check? If you take that away, and so it's we.
I don't know if there is a hard and fast definition there. I think ecosystem engineer might be like a more up-to-date term. I think it's probably got a lot of overlap with keystone species, where like elephants are considered ecosystem engineers because they walk around, they spread seeds, they knock over trees. They're like engineering their own landscape to a certain extent, and they're providing habitat for a lot of other animals. They're like, when they go to wallow in the dust, like they're creating a lot of bare ground that a lot of species of wasp and stuff like that need in order to be able to reproduce. They lay their eggs there in the dust and the sand, and stuff like that. So maybe like ecosystem engineer would be a more up-to-date term. I don't know. We should talk to an ecologist.
I like that. Yeah, it's difficult. One of our mods, Grimley, he's been challenging me on that. He'd be like, well, is this also a keystone? I'm like, oh man, I really don't know. Every time it seems like, but that shows like the interconnectivity of an environment, right? If you take out something that you deem maybe not as important, it actually does have fundamental downstream effects.
For sure. And yet, with going back to the example with the sea otters eating the urchin and preserving the kelp, there was an example where there was like change in activity with fishing fleets, and they were overfishing a certain area, and so these orcas, killer whales, would normally be eating salmon, in this area, suddenly there's no salmon left. And so the orcas go to eating otters. And then they're removing otters from the ecosystem. And then you've got this, population bloom in urchins, and then they eat the kelp, and then the whole kelp forest collapses. So it's like just one change like that can have huge ramifications. And, but yeah, in that circumstance, would salmon be a keystone species? Would orcas be a keystone species? How do you define that? I don't know. I actually don't know. I don't know if there is a hard and fast definition.
Yeah. Alex mentioned the wolves being returned to Yellowstone as like a keystone species being brought back.
Yep. Oh, speaking of which, Balint, did you hear the news about wolves in Colorado?
I have not.
Yeah, I think there were 5 wolves that were just reintroduced to the wild in Colorado for the first time in like 100 years.
Wow.
This is like yesterday or something. And I wish them the best of luck. Holy cow.
Do you have a suspicion of, is there a prognosis of how likely something like that would be with 5? Because that's, naively, that seems like a very small population and almost like a genetic bottleneck.
I know there's been, this is not like, it hasn't been easy to do this, so they're starting off small because there's been a lot of political pushback. Ranchers in particular hate wolves. And they spent like, the ranching lobby literally spent like millions and millions of dollars fighting against this. So yeah, so they're going to start off with 5, and hopefully it goes well, and then they can introduce more so that they can actually like establish a breeding population there, because 5 is not very many.
Yeah, so it's similar to the thylacine, right? Where it's like the ranchers were like putting out bounties and trying to hunt into extinction, and now there's a group trying to clone it and bring it back. But then are you going to run into the same thing with the existing ranchers as you bringing back a species? And it's really sad, right? When you hear these stories.
Yeah. Like people just not wanting to make room for other species. Sometimes very intentionally, and sometimes largely unintentionally, like with all these birds.
Do we have a positive takeaway from this paper, or is it just doom and gloom?
I think, I don't know. I try and think of these things in terms of like, how can we learn about this to improve the future? Having some idea of like the sheer diversity of birds that existed at the time and how vulnerable they were to extinction. Just like understanding that is the first step in trying to make sure that we don't repeat the same mistakes of the past. And I think it also highlights how important paleontology and, I guess to a certain extent, like bioarchaeology like this is. A lot of these birds are not, they're not old enough to actually have fossils. Not old enough for those bones to be fossilized, but this just goes to show that, like, I feel slightly vindicated by this, or validated as a paleontologist, because it's like, this is what paleontology is so good at, is providing context to the world around us today, giving us a deeper understanding of the present and the future based on the past. Because what we have today is just the very tip of the iceberg, There's this 3+ billion year history of life on Earth, and if we don't really understand that, we're not really going to understand the present or be able to plan for the future. And so I think this paper kind of fits into that idea. It's providing some context and kind of showing how important it is to understand the past.
Yeah, I think this figure was kind of getting, as how you were saying too, of like there's an observed number of extinctions, there's some in the fossil record, and there's a ton that are considered to be undiscovered. And like you're saying, like the observed ones, right, are probably too new to have been fossilized.
Oh yeah.
But then there's everything else in between too, and presumably other things that we've killed off that we aren't even aware of yet. And so this number probably is even higher than that 14-15% they were estimating, I think.
Yeah. And shoot, this is something that I'm actually going to be talking about this on Friday. We're doing a special coelacanth stream on my channel.
Nice.
And you're familiar with the coelacanth, of course.
The living fossil.
Yeah. We'll be talking about why that term isn't my favorite, but
this is a dinosaur. Birdie.
I have not.
Birdie dinosaur.
It has not been
dinosaur
kicked out
birdie.
yet.
Birdie, birdie, dinosaur, dinosaur, birdie, birdie.
It's a fun phrase, and I see why it's caught on, but like it's not always super helpful. The point is, the reason I brought up coelacanths is that they are now considered critically endangered, and they were just discovered back in 1938.
Yeah.
Like, we had no idea that this group of fishes had any living species at all. We thought that they went extinct at the end of the Cretaceous, and now some living populations were discovered. One in like kind of between Africa and Madagascar, and one in the Indonesian archipelago. And there could be as few as like 1,000 individual coelacanths of both populations there.
Wow.
And it's like, there could have been other ones throughout the world too that have already gone extinct and we never would have known that they existed. Like in modern times, there's a chance that like there were other coelacanth populations in similar habitats around the world. We have some vague inklings that might be true. There is this really cool like human-made artifact from Central America that's like a beautiful coelacanth, and it's like, how would anybody have made this back in the 14th century if they hadn't seen a live coelacanth, you know? But we've got no indication that any of them live anywhere. Like maybe there's an undiscovered population. It's stuff like this, undiscovered extinctions, you know? Like there's so much basic on the ground work to be done in fields like this, whether we're studying birds, whether we're studying the diversity of fruit flies, whether we're studying other stuff like that. You need to understand what's there before you can actually do anything with regard to conservation, you know? Not before, but like it helps you to do that. There's so much work still left to be done. I think a lot of people don't realize that. I think a lot of people kind of assume that we have an idea of how many bird species there are today or how many there were recently. And like so much of this basic work is it still hasn't been done.
I mean, that's why we like to do, when we do Science News of the Week on Fridays, we always kick off with the new species of the week, because I think it illustrates like, we like, and we fall into this, right? We know everything about the planet, but if there's a new species every single week, and that's just, you know, the 50 some odd papers review. There's a Zookeys journal publishes an absurd amount of new species every week, right? It really humbles you into how small the world is, and then if it's that rate that you're discovering new species, imagine the extinction rate that we're never going to catch.
Exactly. Yeah. And then being able to understand like what is the background rate of extinction? Like how often do species go extinct just naturally due to natural changes that are happening? You know, if we really want to understand the effect that we're having on the planet, we got to understand what that baseline is before we can understand what effect we're having on species and how many species are going extinct because of us. Like that's really important perspective to have.
Yeah.
And I think that's one of the reasons why this is such an interesting paper.
So chat, Friday, on Danny's stream, the deep dive into the coelacanth. Underneath, I can't give him a fancy shout out yet, but underneath the stream title, hit the name and hit the heart button on Danny, please do so. And then
appreciate that. Yeah.
James R. Gurney, how long before
the
TikTok?
Oh, sorry, go ahead.
James Gurney was asking how long before we find a TikTok.
Oh shoot! Well, that'd be very, very surprising. We've got fossils of Tiktaalik. Isn't that enough? But yeah, with the coelacanth, that was the reason I'm doing the stream on this Friday. Is that will be the 1938 to 2023. So that's 70 years. It'll be the 95th anniversary, 85th anniversary.
Oh wow!
85th anniversary of the discovery of Latimeria columnae, the West African coelacanth, East African coelacanth. Excuse me. It was on December 22nd, 1938, that it was discovered, and so we'll be talking about that whole story and why it's so exciting and why it's relevant to our lives today. It's gonna be a lot of fun.
Nice.
Yeah.
So Danny, want to be respectful of your time as always.
Yeah.
Do we have time for the last paper? No pressure at all. I wanted to make sure I know you're hard out.
No, I appreciate that, Balint. Let's do the last paper.
Okay.
Yeah.
So this one I picked because I was a sucker for seasonal stuff, and I wanted something Christmassy.
Nice. I like it.
So, I don't even know how to spell it. Nectadrin B is the active ingredient in nutmeg. And so that's why I ended up picking this paper, because I was like, let's do something Christmassy before we have Christmas next week.
So that was the idea behind this. I like that.
That was the weird hook that I had here, right?
Mm-hmm.
It's also a pretty new paper. It's published November 19th, so about a month old. And so these studies are always, part of which frustrating to me, and the other part very interesting to me. And the reason is, I don't know if you remember these papers, Daniel. They came out a couple of years ago on if we give an organism Splenda, they get cancer and die. And so it was like an argument against Splenda.
Mm-hmm.
But it was if you looked at the paper, the concentration that you would need of the Splenda was like you'd have to be shoveling Splenda in your mouth every day.
The creatures were being, physically crushed to death by the weight of the Splenda.
Exactly. And so it's like
practically.
It was one of those where it's like at any concentration, eventually something will take you out. And so it was.
Yeah.
But the nice thing about this kind of study is that it's the opposite direction. It is not a molecule that kills something faster, which I think anything could do. It makes the animal live longer.
Which is, that's a much more significant finding there, which is why this is so cool.
Yeah.
And nutmeg of all things.
Yeah, I wish there was some kind of... I mean, the background study, like, the information that they had was, like, in the literature, nutmeg has shown to have antioxidant properties. And that we know antioxidants pull out the oxidants, right, in the bloodstream, which can cause DNA damage and cause cellular aging. And so it can slow down aging. I wish, though, there would have been some kind of interview or backstory with the authors of, like, how did y'all actually come up with the idea sitting around the table for nutmeg?
I like that. Yeah.
And so
they should do like a promotional video for their lab. That would be interesting.
Right? I kind of like when there are interviews and you get a little bit of the background of how exactly did the experiment work? Because for those in chat, it's usually not based on the figure order, where it's nice and clean. There's this hypothesis and an observation, and then you go into the genetics. Hanny, I would love to have it be like they were making pies for Thanksgiving, and this one painted fly kept hanging out for like the next 4 weeks, and it just wouldn't die. And so they're like, you know what? Maybe it's eating the pie, or I don't know, like. Like with Lita's PhD work, it was we were sitting in the lab and one of us goes, wouldn't it be weird if this thing happened? And then we tested it. Like that's how that whole started. Like that was the hypothesis going in.
Yeah.
And I think that like that little backstory is kind of cool. It humanizes the science. It doesn't make it sound so regimented and we always know exactly what we're doing and we're just this infallible ivory tower and. So
there's a lot of guesswork and a lot of like, wouldn't it be weird if, or what if this works that way? Like, that's the same way in paleontology too, for sure.
And that's the cool part. Like, I think it's okay that science is like that. It should be a puzzle where the pieces aren't fitting great, and then all of a sudden we accidentally get them to fit together, and then you're set.
Yeah. There's like that famous story of old Chuck E.D. Charles Darwin. When he was on the Galapagos Islands, and then a monkey fell out of a tree onto his head and gave him the idea for evolution.
Really? I've never heard this.
No,
I was like, "What? Really? This is insane."
No, I took that. That's a joke. That's an old joke from The Onion.
Okay, I was like, "Huh."
But yeah.
I like that.
I thought it was funny.
We actually had Danny a paper covered by The Onion.
Oh really?
Yeah, that was one of my like life highlights.
Is it one of those ones where they have the different people's photos and like their response to it?
Yep. It was
nice.
Fruit flies
You'll have
laying
to send me
on.
That. I'd love to see that.
I'll send it over. It was like fruit flies laying on alcohol laden food to protect from parasites, and so the people were saying like drug flying like. as long as they're, buckle up or they don't take someone else with you, like that. And we were just like, did we just get featured in The Onion? What is this?
That's great, and I thought it had to be that because that's like one of the few times that The Onion actually uses real headlines is when they do the like, let's ask folks on the street about this. Yeah.
And so JD, you have a silly question, but an important question here is that I'm not saying that pumpkin spice lattes extend lifespan. So this is the difficult part of the interpretation of the data, right? So I'll show you in a minute like what they actually did to demonstrate that there was a lot of anti-aging reversed. But how you interpret this to humans, like I'd love to tell you to go buy a nutmeg extract pill and make yourself live longer, but it's not clean enough evidence to do that yet, right? So I think there has to be a lot more testing, concentrations, metabolism of a mammal is different from an insect, and so it's something where like the popular news media. Oh, Alex may have found it. Alex Vixen may have found it. Yeah, Alex Vixen found it.
Nice.
And yeah, it's the pictures of people down below. Thank you.
They always use the same photos too, which I think is really funny.
Oh yeah, it's the same photos every time.
Yeah.
So yeah, I just think it's an interesting component. Like in the popular media, even in this article, we're already kind of harping on the fact that, like JD, it was almost essentially like, a chai latte or something like that can help you live longer. The holidays, I think one article, I mean, it wasn't even joking. It was like the holidays shouldn't stress you out about doing badly. Just have some nutmeg. And it was, there's already this desire.
What they should really say is, if you want the, like a lot of parents who probably got your child, a fruit fly as a gift for Christmas. A new pet. If you want that fruit fly to live for a long time, give it some nutmeg. That's practical advice right there that everyone could use.
So, the other reason I wanted this paper is kind of building on what we've talked about before, right? We've talked about aging studies. And remember, it was like 20-day-old flies that was the old flies in that previous study. And I thought that you might find this cool because they actually went out to over to 200 days. So they did like whole lifespan studies in this one. So it's kind of getting more at what is like the extreme end of it. And later on they had age groups 90-day old that they were testing behavior on, right? And so that's much older than that 20-day old that we were looking at. And so I just thought that was kind of cool that they're trying to push now of like what I would consider a much more of an aging phenomena rather than just your small windows. It seems more extreme to test something at the end of something's life, right? Versus just in the middle of its life. So I thought that was kind of building on what we chatted about before. And looking at whole lifespans, like they did this to males and females, and they find that both have resistance, like with the nutmeg. And so here, and I like Corn Relish says chai lattes have definitely made me live longer. But yeah, like the black right here, this, and I don't know how easily y'all can see it, chat. I have a lot of difficulty telling these colors apart, but the black line is the one that's closest to the y-axis bar, and so the y is % survival, the x is time, and so if you're not fed any nutmeg, everyone's dead a little after 100 days. But different concentrations of nutmeg and the active ingredients in it extend lifespan, and so this RAP is something that's been shown in the past to extend lifespan, so that's a positive control. And then this NEC-B is what they're getting extracted from in the nutmeg, and they're finding that also seems to extend lifespan pretty well. And it's both male and female. And we talked about this too, Danny. This Oregon R is one of their wild type line that was identified in Oregon, and the R means it has red eyes. I don't come up with the naming system, right? But that's their wild type. It
starts with an R,
yeah.
Red.
I never knew why it was, as an undergrad. They're like, oh yeah, we're gonna use Oregon R, and I was like, oh, I thought it was like Oregon R, because people like said it so fast. and it was known as Oregon R. And then Murph points out too, it's interesting too, the concentrations, like the 50 and the 200 look pretty similar. So the higher doses for the nutmeg, it seems to top out, and that's seen in other organisms as well. Like at above certain concentrations, it's like it's not returns. Yeah, it's like almost not benefiting anymore. If there is, it's so slight that it's not statistically significant. Or Alex, day 207, the fruit fly AS54013 is still alive, showing distressing behavior as it's figuring out how to escape and get enough to the nutmeg stores. Exactly. Timmers, as I try very hard now to say Oregon correctly, because I thought it was organs for a while of the fruit flies. I was like, what about it? Is it organ? I don't understand.
A lot of people also say Oregon, which that's not how people from Oregon say it. I'm from California, so I've met a lot of Oregonians in my life. But we say Oregon here in California, too.
The appropriate
kind of
way.
Like Nevada versus Nevada. You know? It's Nevada if you live in Nevada, or if you live in California. But it seems like everybody else seems to think it's Nevada. But, yeah.
I think in the South we always said it Nevada. But it's because we threw that twang on the ah.
Yeah. Okay.
And so that could be... they might not like that either. And then there was this interesting component as well. I thought you might find this cool, Danny. It's these flies called the DGRP lines. So these are there were 200 flies. They were caught in a market in North Carolina. So the researchers went to a farmer's market that had fruit, and they caught 200 female flies.
Wow.
And they made what is called an isogenic line. So they put each fly into a vial by herself, and under the assumption that she's already mated.
Okay.
And then they took the offspring and inbred them and did it over and over and over again until the idea is that you're getting rid of genetic variance between the individuals, and that each of these vials of 200 individuals now they're genetically identical, but 200 very different individuals.
Okay, yeah, I get that.
And so then they sequence the animals, and then the cool thing now is, the idea behind it is you can do a test of any phenotype. If you test a subset of them, whether it be or all 200, you should get a range of phenotypes. And in this case, it could be aging, but the idea is then you can put your data into this computer program, and it'll spit out for you the genes that are associated with your behavior or whatever phenotype you get.
Interesting.
So like
that's really cool.
So like long live, for example, like they did some on lifespan, not in this study but in the past, where it's like, let's see, these animals usually live to 250 days, these ones live to 60, and then we have some in between, and you put it in this program, and the program's like, okay, based on the sequences and the differences, these 10 genes could be involved. And then you go manually knock out the genes or turn them up and off and see if you can change the lifespan of a other fly. And you know, it's a way to do a screen for genes that's supposedly not as difficult as if you did like mutation and tried to figure out how things are different.
Gotcha. Okay.
And so they did this for DGRP flies, and as an effort to try to see like what again is the genetic basis for this. So they showed, you know, two different, the DGRP line number 100, which is, this is how they're identified as just like this it's a Drosophila genetic resource panel, and then by a number of which one it is. And then you just, you know, do your usual graphs, and then you put in the data of whatever you want to look at in the program, and it spits out potential genes.
That's really cool. And it gives me a question too, maybe a bit of a tangent, but you say this with like such familiarity here. You're like, oh yeah, like an old friend, like DGRP 100. Is this like a strain that's used in a lot of different laboratories and stuff like that?
So the DGRP lines themselves are used in labs that if you want to do like a cheap experiment, because the thing is that if you do an old school mutagenesis, if you mutate the genome and then you try to do the same thing, you might find a really cool fly that, let's say, has extended lifespan, but unless you
Sure.
sequence its genome, you
don't
have
really know
no
what's
idea.
going on with it.
Exactly.
Yeah.
So
it could have like a novel mutation just at one individual or something.
well, and then the likelihood too that there might be, there's not one mutation in that individual. There could be a ton of mutations, and so you
Sure.
sequence the genome and try to figure out, is it a combination effect, or it gets really dirty and it gets expensive fast. So we had a lab across the hall from us that we jokingly said we're looking at the brown butt phenotype of a fruit fly. So on the tergite sections, there's an amount of dark pigment, and this one graduate student was measuring the third segment of a tergite and the amount of percentage of brown that it had in terms of melanin, and did that for all 200 lines and figured out what gene is contributing to the brown buttness of that third tergite. When I was an undergrad, we had a postdoc do this with immunity, and the idea was, if I infect the larvae of this fruit fly with a parasitic wasp, what percentage of these are successful immune responses? So how many times does a fly hatch out versus a wasp? And then if you put that data in the program, maybe it'll give you new immune genes. Turned out that did not work. It turned out that was too complicated. It turned out there was just too many factors at play. So you have to keep it simple for this to work well. And so something like lifespan is, I suppose, simple enough, but a host pathogen interaction is too complicated. At least 10 years ago it was. We also tested it for memory as well, and that seems to be working pretty well. But yeah, it's like you have to be careful with complexities of behavior, because sometimes it's just it can't delineate between the
makes
differences.
A lot of sense.
Yeah.
So
there are some that are more commonly used in studies if you don't go for the full 200. And honestly, so here's, Danny, I would love your opinion on this, because it's probably actually a confounding variable. These X numbers do really well in a lab, and so they're easy to grow up and get a lot of.
Okay.
But presumably those are still genes there that are influencing the experiment, because they're just going to be doing well in a lab setting. And so are you actively selecting for something there?
Right. That's interesting. Yeah, where it's almost like you've domesticated the fruit flies to a certain extent, and interesting. But that's what you got to do, because that's how else are you going to do the experiments? If you have really, really finicky fruit flies, it's not going to work.
Which is weird, right? You'd assume that a fruit fly is just going to behave, but no, there's a lot of cases where they just don't do well. And it's like, is it the right food source? Is it the light dark cycle? It's just. But yeah, like Timmers was saying, it's easier than cutting a rat or a mouse in terms of financial for a screen, testing it on mice. Like, that gets so pricey so fast. And if you don't look at the right tissue, that's a whole other mouse you have to sack and go undergo this experiment. And with a fly, if I don't look at the right tissue the first time, I just age it again, and then we look at it, and it's a month, 2 months at the most, and I can repeat the experiment. And the timing and generation time is super important. So it's just cool that there was this DGRP here, I thought.
But to clarify though, have you worked with DGRP before?
Yeah. So I've
so the 100 there, is that like a particular strain?
Yeah, that's one of the strains. So it's
gotcha.
it's that 1 through 200, and there was 100. It was there was something in the 40s, the 80s, and I think the 170s that we had selected for. That were recommended to us by other people who worked on them a lot. These are really healthy and they do well in the lab, and and there's also, it seems like there was but the numbers themselves don't mean anything in terms of where in the genome they're different, because it's going to be based on phenotype. And actually a graph that you get out after analysis looks like this curve. There'll be if it was lifespan, you'd have low short-lived ones and then up to high-lived ones and stuff in between. So you really want this really smooth curve, that you can then
Right.
do a comparative analysis across the curve and see, okay, well, what genes are different in this population versus this one, and in the intermediate, does it correspond to intermediate levels of the other genes? It's a cool system. It's just, it seems finicky. So a lot of
Yeah,
people use it
I
for
get
like the option for natural, quote unquote, natural variance, and asking that question.
Interesting. And so with these different fruit fly strains, like DGRP 100, this is like a continuous unbroken lineage from the time that those flies were collected in the
At that
the
farmer's
wild, right?
market, yeah.
Which was what, like 20 years ago, something like that? Probably longer?
2010?
2010.
Yeah.
Oh, okay.
I believe 2010. It was in North Carolina at a farmer's market. I think it was UNC where the research was done in Trudy McKay's lab.
Nice. Okay.
And I remember our lab when I was an undergrad, we were excited because we got access to the flies ahead of time. So like we got them in the lab before some of the other. But it was like, you can't publish on them. Like she was like, you can do experiments on them, but you have to wait for us to publish the paper. And so it was like, that was a cool sharing of resources and information, and it obviously made sense of when to wait.
Yeah, and they trusted me with that too. That's also cool.
Yeah.
Yeah, it wasn't all about
Well, because there's plenty of that are welcome dagger. JD, sleep well.
Yeah.
JD, sleep well. Thank you for the saxophone.
Would you happen to know what the longest running strand or strain of are they called strains? Do I have that right?
Yeah.
What's the strain that was collected earliest from the wild? Like, what is the longest running numbered strain like that of fruit fly that are still used in research?
So Canton S is was identified in 1898.
Holy cow!
And then Oregon R was early 1900s. Those two.
So that's just a continuous populate. Like, they've been generation after generation kept alive in laboratories and then used for research like this.
And you know what's wild to me is they still have innate behaviors. For example, like you said, if that fly has been strain in the lab since, let's say, 1900, and you're breeding them, breeding them, they haven't seen the wild for over 100 years. And yet if you show them a predatory wasp that they've never seen before, they still have behavioral and genetic changes that accompany that.
Yeah, that makes a lot of sense, because you're not going to be selecting against that. Yeah, I would just wonder how much genetic drift has taken place since then. Like, after these probably thousands upon thousands of generations. You're not going to have a lot of active selection, I'm guessing. But like, how much genetic drift took place during that time? Did they have preserved? Okay, here's a question. Are there preserved specimens from like the initial generations of these strains, like that you could go and actually sequence that genome or whatever, or even look at them phenotypically? Like, is there a drawer full of little taxidermied Drosophilus somewhere?
So like in ethanol tubes, so you can still extract the DNA from them. But so it gets wild on multiple levels. Okay. If you take care of these Canton S fly, like if I give you some Canton S and I get some Canton S, and we take care of them in our labs for the next 10 years, they're not actually going to be the same after 10 years, because you and I take care of them differently. Yeah. So it's already a fallacy of,
for different things.
Yeah, it's already a fallacy of what is a control. Like, your control is not the same as my control.
Right.
As just a feature of drift. But there is this thing called a transposable element. It's a hopping genetic element that can insert itself in the genome, pick itself up, insert it elsewhere in the genome, and it could be very damaging, because when it removes itself or excises itself from the sequence, it might not be a perfect excision, and it could take regions of the genome with it, so it can disrupt a genetic sequence in that way, or it inserts itself into a genetic element and breaks that structure. In Canton S and Oregon R, which we got before the 1950s, they have no genetic repressors of transposable elements. So if you give them a transposable element, it trashes their genome. That thing just hops around and it just annihilates them, and they are unable to pass on anything.
Wow.
But modern, like wild caught ones, if you do that same experiment, if you give them this transposable element, it hops around a little bit, and then all of a sudden there's repressors that come in and turn it off. So something has happened in 50-ish years. Astroeusis, thank you for the follow. In 50-something years, that has evolved in the wild that's not in your domestic population.
That's really interesting.
Yeah.
And they just have no defense against that, huh?
And so that's... when I heard that, I'm like, that is insanely cool. So that is one, I think, big difference, where that would also tell you that at least in wild populations and domestic, there's a big difference, and presumably the evolutionary difference is over time. Like, pressures have been different as well.
Yeah. That's also really interesting to me because it implies that there's not a lot of risk of accidentally breeding a strain of super Drosophila that, like, if they get loose in somebody's house, then they're just super comfortable living alongside people, and they'll just take over the world because they've been bred to live under fluorescent lighting and blah blah blah. They just would not do well in wild circumstances.
No, it's crazy, right? Yeah, like if you let most of them. So the way that you know that you've inserted a genetic element is that you make it in a white-eyed fly. That's like a mutant that was identified by Thomas Morgan in 1911. And when you put in a gene or disrupt a gene, you attach to that disruptor the wild type copy for red eyes. So if it's integrated in the genome, this new animal also has red eyes. It has that other mutation it's carrying, but now it has red eyes. So it's your marker that your genetic insertion worked.
Right, yeah.
But it does seem like a lot of these are sickly, or there's a lot of issues with these animals. So if you release them in the wild, they wouldn't make it.
Interesting.
I had the same thought, actually, like when I went into like a genetics lab, like, oh, can we breed the superfly? And they're like, yeah, no, it unfortunately, like, there's so many like checks and balances when you're trying to interbreed some of these mutants that because some of them are made with the genetic background, like in either an Oregonar or in Cantoness, you might combine genetic elements that are incompatible, that aren't like your mutations that you're adding, but just something in their genomes. Like, I guess I think of it like if you have two patients, both are carriers for cystic fibrosis, they themselves have a lineage that's okay, they're just carrying, but then if they mate...
Because it's recessive,
Then, yeah, exactly. So you'll start bringing out phenotypes that you wouldn't account for before. And so, yeah, it's a cool study, yeah.
Interesting stuff.
Yeah.
Shoot.
And so, yeah, this was just a cool, like, Hanny was asking about the aging for this. They actually did the climbing index that we were talking about earlier tonight as well, with the negative geotaxis, and you were asking about, well, how do you tell if the animal is just aging or like locomotor behavior? So they looked at, how quickly and how far they're going up a particular tube. It's super easy. Like, you take a tube, you get ticks along it, and then you count how many individuals are in a given zone after a set amount of time, and that's how you take it. And so, we can see like these ones that are fed the nutmeg, and then the control substance are higher than your non-antioxidant fed individuals, both in males and females, as well as for the DGRPs. Body weight was also an interesting issue. I didn't remember that this was something in flies, but as flies age, they increase in body weight, much like humans, right? As the older population, because you're less mobile, like there's a lot of factors at play. But feeding them this nutmeg, like in the dark bars, all of a sudden it goes down, and it kind of corrects that.
The weight goes down?
Yeah. Which is counterintuitive, right? You would think, naively, I would think that down weight would be a health detriment, but it seems like,
Well, that's funny, because you say that, and I would think it'd be the opposite for a volant animal, for something that's flying. I would think that an increase in weight would be a bad thing across the board. But I don't know. Yeah,
Yeah,
that's interesting. I wonder, is there any indication that nutmeg does something to kind of reinvigorate the metabolism or something, and make sure that they're processing food into energy more efficiently or something like that?
Yeah, so they didn't do metabolomics, but that would be a neat
Okay.
next step to look at. They did look at eye cell morphology. So these are a bunch of fruit fly eyes, and they're trying to see, like, is there an increase or decrease in damage over age? And so you're looking at 30, 60, and 90-day-old individuals, and if we zoom in here, there's your 90-day control fly, and it's got all these dead cells along the bottom. These next two are your control, like fed individuals, which are already a known antioxidant. And then here's your nutmeg. You can still see like a dot or two.
Yeah.
But it's nothing like when the entire part of the eye is dying. So this is like their proxy for the nervous system is better protected.
Right.
They didn't like. They also did look at brain structure as well, and this is actually it's a little bit of my graduate work too. These are cross sections similar to like this 3D print that we've got here. But as the animal ages, holes emerge in their brains, just cell populations dying, and so you can see here, like, this is a control male brain, control female, and there's all these like empty regions that you're seeing. But then if we scroll over to the ones that fed nutmeg the same age, like the cellular structures are in place.
That's wild. Yeah.
And like this to me was like the really shocking one. Like, I'd love to see some sequencing on here to see if like the metabolomics genes have been turned down in like a lot of tissues in the body, because it seems like it's acting everywhere, right? It doesn't seem like it's only in the brain or it's only in the gut. It's like, or muscle. It seems like muscle, eyes, like, so cellular age of all over, brain tissue. And so how all those, like, where is it actually acting, correcting? Because it almost reads like a universal corrector.
Interesting.
And so, and Hanny, what's suspected is that you're just delaying the aging process. So there's a mutant actually named after the biblical character in Methuselah, the longest lived human. And so those fruit flies normally live for, instead of 90 days, it's like 120 to 150. And it seems like they just keep aging, but it's at different rates for different tissues. And so it's not necessarily like they're a young individual. It's just you're just allowed to live a bit longer because the aging process is a little bit different. So, yeah, it's just you're living. It's and also, Hanny, we can't ask the flies, right? If they're like in that youthful state longer, that you can ask if we did sequencing, if we did comparison of the genome not the genome sequence, but transcriptome. If the 60-day-old nutmeg fed fly looked like the 25-day-old non-nutmeg fed fly, you might have the profile of a younger individual, but versus like behaviorally or something like that would be a little bit harder to demonstrate, I think. And yeah, Timber is like what this means for Alzheimer's or multiple sclerosis or any kind of aging. This is where it gets really exciting, but it also gets easy to jump very far ahead, right? Of like what is the correlation with humans? Because we are close, 70% of the genome is identical to a fly, but
we do look a little bit different,
Yeah, occasionally. Some people do. But no, it's like where do you draw the line? Like, where do you get not too excited about this and try to automatically, like, I'm gonna go out and just gorge on nutmeg.
So what would be your next step for continuing this, Balint? What would you recommend to these authors, or what would you do yourself?
If I had all the money in the world, I wanted.
Someday, hopefully, you will.
It'd be No, Danny, you and I are gonna hold the SciComm symposium conference. That's every year. It'll be the biggest one in the United States and then worldwide, and we can chase the dream of being science communicator. That's it's gonna happen, Danny.
I look forward to that.
But no, I would do the sequencing experiments, the transcriptomes across each of these different tissues. So you've demonstrated eye cell, brain, muscle. I would do fat body as well, so that's the side of primarily the metabolism of the organism, and test at these different ages. Run those sequencing experiments to see how they're different. And what I would predict is that you should see the older nutmeg-fed individuals have the similar profile as a younger non-fed one. And then you might be able to see what genes or gene families are changing along with the feeding. Like where is this molecule acting? Right, like what gene cluster is it affecting? And then, to me, I want to go in and then genetically alter those genes as well. So in the absence of feeding them this compound, if I just mess with certain genes in the same direction that they are in this profile, they should then result in the same slowed down aging, right? Like I want to see what that mechanism is, like what is being acted on. And then, presumably, I hope there would be a conserved gene in mammals, and we can test it in mammals too, because one thing that's going to be hard, right, is dosage. Always is going to be dosage. But if you can go into a mammal and change that in a mouse, that gene right away, and you get an extended lifespan, then you can be like, okay, at least we know that the gene itself that's targeted works. Let's see if nutmeg acts on the same pathway, and then we can start doing a dose response curve. Because I found, like, we did some dose responses and attempted to start doing stuff in mice in grad school, and it spirals really quickly in terms of price and time. So it's almost easier to have, like, the mechanism figured out first, and then go to the small molecules that you're trying to get to affect the behavior or phenotype of the organism.
Gotcha.
So that's what I would do. Proteins as well.
Cool.
Proteomics and metabolomics you can do too. That's going to be a bunch more in price, but that I think could also help inform if you really wanted to go above and beyond. This isn't the journal Aging, so that's, it's a pretty reputable journal, but it's not super
high profile.
Yeah, it's fluctuated. But yeah, like even like these neurodegeneration indices that they're looking at, like, these animals at 90 days old fed this protective holiday nutmeg are near undetectable in terms of how, like, the lower score, the lower the neurodegeneration that's occurring, which is just crazy to me. The giant decrease between, like, these certain levels.
Yeah.
Yes. So, they did the locomotion assays, the body weight, the eye imaging, the histological analysis of the brains. I would do the sequencing next, metabolomics and proteomics if we can, to really figure out, like, what's the mechanism, and then try to adapt it and mess with the genes ourselves and see if we can replicate it in the absence of the molecule.
Gotcha.
But I thought, cool study. We got to do, it's a holiday cheer, and then it has the DGRP that I thought was a cool phenomenon. And it I was trying to be a sucker and, like, link it back to earlier stuff that we've talked about with the aging phenomena.
Yeah.
So I don't know. I would be like, I thought it was cool. Again, it's not like a hard hitting, like, change the world paper, but it could lead to something.
It's interesting stuff, and I'm eager to see if anything comes out of this nutmeg. Interesting. Thanks for picking this, and thanks for sharing your expertise, Balint.
Sir, thank you for coming on and sharing your expertise. This is awesome. I hope we keep continuing this into the new year. It's seriously become a highlight of my week. Like, I love reading papers that I'm like, it's reading it different. I'm like, what would Danny also think is cool? And, like, us talking about, like, new things. And I'm telling you, that those papers, especially the one with, like, here, like, the degrading of the plant matter and, like, linking it back to the past. This is just so cool. Like, I really, really like this paper.
it was a really, really cool talk, and I'm gonna see if I can actually bring the lead author on as an interviewee on my channel eventually.
Nice.
Because she's super cool, and this sort of thing is right up my alley. Like looking at modern organisms as a key for extinct organisms. That's kind of what I'm all about. So like most of my research also is kind of along a similar plane. So it's just cool stuff. I'm glad you liked it too.
No, it was amazing. Yeah, thank you so very much, Danny, for coming on. I know you've got an evening presumably planned of high intensity, high octane fun.
You go to bed before too long.
Hey,
but
I feel you on that.
Yeah.
But guys, please go follow Danny if you aren't already. Single greatest science communicator on Twitch. The reason we are here, the heart button's at the top of the chat right now. There's 292 of you here. You know, if you haven't clicked that heart button,
holy cow, there's a lot of folks watching.
sir, I didn't want to tell you, but I got this image from one of our mods. We're actually on the front page of Twitch.
Are we?
We are.
Well. Hello, front page. Excellent.
So we've been, you know, highlighting the beauty of science on the front page of Twitch, which I think is really freaking cool.
That is really cool. Yeah.
That we have this opportunity, and Danny, that you're on with us, and us, you know, hopefully, like, in my mind, you know, one of us does well, the rest of the scientific community here does well, too. And thank you so much for coming
on. A rising science tide lifts all labs, right?
It lifts all labs, exactly. But seriously, thank you so much.
Yeah, you too.
Next week, if you're available.
Yeah.
Perfect.
Sounds good.
Awesome.
Right,
Thank you again, Danny.
Now. My earbud is the battery on it's dying.
Perfect timing, then.
About to turn back into a pumpkin. Yeah. I'll see you later, everyone.
Thank you, Danny.
Take care. Goodbye, everybody.